Good morning, sir.
Good morning. Good morning.
Good morning, everyone. I, Ms. Anupama Tomar, on behalf of Department of Microbiology and Biotechnology of Patkar-Varde College, welcome you all for one-day Rusa-sponsored national webinar Biofacet. It gives me immense pleasure to welcome our guest speakers of the day, Dr. Giridhar Parvatam and Dr. Jairam Reddy. I will now give a brief introduction of our college. Chikitsak Samoos Patkar-Varde College was established in 1964 in Mumbai's western suburbs. Since its establishment, it has constantly worked towards students and society welfare by bettering its infrastructure. Our college is an autonomous college affiliated to University of Mumbai and has been reaccredited with A plus grade by NAC third cycle with an institutional score of 3.53. We have also received Best College Award in 2016-17 from University of Mumbai and are a DBT Star College awardee. We have also obtained ISO 9001-2015 certification. The college has grown steadily over the last 5 decades, and in keeping with the spirit of learning and growing through criticism, we have set high standards, working continuously towards advanced goals. Our college offers a wide range of undergraduate, postgraduate, and PhD programs. In addition to the traditional programs, the college also offers the emerging need based programs like BSc in Biotechnology, Microbiology, Aviation, Computer Science, and Information Technology, BMS, BCom Banking and Insurance, BCom Accounting and Finance, MCom Advanced Accountancy, MCom Banking and Finance, and MCom Business Management. Our students have consistently bagged top positions in university and board examinations. A brief introduction about our department. Department of Microbiology and Biotechnology was established in the year 2006. The department has been organizing Biofacet every year. Started in the academic year 2012-13, Biofacet is a stride taken by the department to provide a platform to scholars, teachers, and experts from various fields of science and technology for academic interactions, elevation, and enrichment. It has also taken initiatives in helping students in extending their minds to diverse fields available in biological sciences. This year, we are extremely glad to organize Biofacet on national level. This would have not been possible without your support and participation. Thank you very much, dear participants. This year, we have organized Biofacet on the theme that is advances in plant research. As we all know that plants are important to mankind from the earliest study of life processes, and they are central to scientific study. Research on plants enriches our intellectual life and adds knowledge about other life processes. The results of research on plant systems also teaches us how to approach problems in agriculture, health, and environment. And to elaborate more on this, we have the first speaker of the day, Dr. Giridhar Parvatam sir. Now, I take the opportunity to introduce the first speaker of the day, Dr. Giridhar Parvatam, Senior Principal Scientist and Professor, Plant Cell Biotechnology Department, CSIR, CFTRI. Sir's areas of interest include plant biotechnology, secondary metabolites, nutraceuticals, food bioactives, and bioavailability studies. Sir has 9 patents and 2 process technologies to his credit. Sir has published 185 papers and authored 15 book chapters. Sir has also guided 12 PhD students and 35 MSc MTech dissertation works. Sir also received international fellowship from DAAD Fellow Germany 1995 to 96 and Boycast Fellow France from 2005 to 2006 and has a long list of national fellowship. Sir has also been elected as committee member for Plant Tissue Culture Association of India and National Academy of Sciences, Allahabad. Sir is also a BOS member in botany at Kavempu University and University of Mysore. It is indeed our pleasure to have such an eminent speaker with us today. Before we begin with the webinar, I request all the participants to be kindly patient if any technical problems arise during the webinar. We will try to resolve all the technical glitches. Kindly post your questions in the chat box. It will be answered at the end of the session. We are sure that this webinar would certainly complement to your learning objectives. Now, I would request Giridhar sir to address the webinar.
Yeah, thank you very much for the nice introduction. First, I would like to share these slides. Let me share. Are you able to see my slides?
Yes, sir, it is visible.
Okay, thank you very much. Okay, good morning to all of you. Thank you very much for the nice introduction. It's my pleasure to be part of this national webinar. At the beginning, I would like to express my pleasure and happiness for the invitation extended to me by the Sir Sitaram and Lady Shantabai Patkar College of Arts and Science to deliver this national webinar talk on Biofacet 2022. The topic what I have selected for my presentation is advances in plant tissue culture technology for food value enhancement. As you know that there is a great demand for plant tissue culture since this the audience gracing this biofacet mostly from the degree colleges and the university faculties. I have structured my talk in general aspects of plant tissue culture, its importance, and the theme is with reference to the food value plants and the food value metabolites. Since I am from the Food Technology Institute, so whatever experience I have gained in this particular domain, that I would like to share rather than going into the intricacies of plant tissue culture aspects. So, if you look at the global plant tissue culture market, there is a huge potential in this area. This is the latest figure between 2021 2030. That means the next coming one decade, the statistics indicates that there is a huge potential with CAGR of 8.5% in this plant tissue culture area, and the market may touch $895 million by 2030. actually, as of now in 2020, it was there around 382.3. That means it's more than double, and anticipated growth is expected. Of course, the tissue culture is not new to the plant scientists and also to the researchers. For the past 6 to 7 decades, extraordinary developments have happened in this plant tissue culture area, and the North American region listed top in developing the tissue culture plants and also utilizing the technologies based on the plant tissue culture. Nevertheless, India being the one of the fastest growing economy, our country is also having a very vital importance and lot of scope is there to gain leads in this area because the Indian researchers are highest contributors to the plant tissue culture rather than the rest of the world. So if you take whatever type of plant systems, the Indians have done fantastic work by contributing in the plant tissue culture area. This is just a picture that gives the importance of a plant tissue culture current market. This is by crop wise. Of course, it's not exact statistics, but the trend it gives. If you look at this graph, you can see that floriculture plants occupies the top priority, top rating and demand in the market. This is the red color one is the 2020 and the anticipated growth by 2030. So the floriculture plants are having the lion's share here, followed by the fruit plants, especially the banana plants are having expected to have a great demand by 2030. Next, wood producing plants like a teak or any oak or like that. Such plants are also organized way cultivation is happening for commercial purposes, followed by the rest of the other fruit plants other than banana plants, vegetable plants, ornamentals, and other things are there. And this is by this slide gives a holistic view of this the next 1 decade, how the plant tissue culture market by stage of development. Here we can consider the 3 different stages. One is explant preparation and inoculation, that means at the laboratory level, then followed by the multiplication stage, and next finally the hardening stage. Rather than the first and the last stages, the multiplication stage is very important. Once you are able to achieve mass multiplication of the desired plant at the laboratory level or at the industry level, then directly it will be purchased by the people who wants those things and take them into the greenhouses for hardening and transplantation. So there is a huge potential for mass multiplication stage also. And this picture gives how the plant tissue culture market is envisaged for the next 1 decade by plant tissue wise. That means specific is wise, types wise. Annual plants, biennial plants, and perennial plants. We know all these the difference between the annuals, biennials, and perennials. So compared to the biennial plants, there is a huge demand for this annual plants in the 2020, and the same proportionately expected to increase the greater demand for this one. Next is followed by the perennial plants. So now let us look at, just I will give a glimpse about the advantages of a plant tissue culture. Even though the faculty members are familiar with this one, just for sake of students, I will give a glimpse. One big advantage is you can grow the new plantlets in a short time in a controlled conditions. Similarly, only small amount of initial plant is required. That means you may need 1 square millimeter leaf explant or 1 square millimeter hypocortical explant to induce the plant tissue, whether it is the callus or direct organogenesis. And whatever new plants what we get are mostly like the parent plant. So the variations will be very less and you may get disease-free plants, virus-free plantlets. And the process is not dependent on season. Suppose if a plant is a winter-prone plant or summer-prone plant in outside environment, you have to wait for the season. But here, since we are doing in the controlled conditions, so there is no need of worrying about all the seasonal influence. And you need only a relatively small pace to perform. That means within the structured laboratory conditions, in a micro bottles, you can do this micro propagation very efficient in a high pace. So you can save the infrastructure related aspect and space constraints also. So on large scale, the tissue culture process helps to supply consumer market with new subspecies and variety. Suppose if you induce any variation genetically in the tissue culture itself, you are able to produce the varieties, new varieties also. Similarly, it assists producing several novel varieties of crops through protoplast fusion and genetic engineering. So, in general, breeding, the commercial, traditional breeding methods, it takes years, but here in a short time, you can get produce new varieties. For every technology, if you don't mention the disadvantages, it is the technology is flawed. So every technology will be having both advantages and disadvantages. Even though the advantages are many, there are one or two couple of disadvantages for the tissue culture also that we need to look at this one. The first one is the tissue culture can require more labor and cost money, and also cost money. That means cost wise, unless you adopt innovative methods, the cost will be high compared to the traditional methods. And there is a chance that the propagated plants will be less resilient to diseases due to the type of environment they are growing. As long as it is in in vitro conditions and greenhouse conditions, they may be disease resistant. But once it adopts the outside environment, again the environment plays a key role. So there are chances to get the abnormalities. And also it is very, very imperative that before being a culture, the material is screened. Failure to pick up any abnormalities could lead to the new plants being infected. This is what just management. Similarly, while the success rate is very high, if the correct procedures are followed, success with the tissue culture is not a guarantee. There is still a chance that process triggers a secondary metabolic chemical reaction. That means within the tissue, because for example, you take callus, consider callus tissue. It is a homogeneous mass of cells. Even though we anticipate organogenesis from this, sometime under some media conditions or whatever may be the reasons or genetic variations, if any harmful metabolites produce, and the tissue may die and you may not get response. So these are some minor disadvantages that cannot be ignored. So what makes plant tissue culture so great? How it is different from the traditional methods? Just now what I have explained, it can be used in the reproduction of a wide variety of species and has more practical applications. This is evident. Similarly, genetically stable plants, we can get it. If you design a new variety, you can alter at gene level also through transgenic technology, and through transformation methods, you can do it by employing the tissue culture, wherein tissue culture will be a tool rather than for propagation. Can be used to promote the survival of rare plant or endangered plant species. That means some many plants are becoming endangered, especially medicinally important plants because of over exploitation. Such plants you can rescue, and through tissue culture technology, you can multiply and propagate. The tissue culture technique relies on the plant innate ability to rejuvenate cells quickly. That is called clones. So it is not the media ingredients. It is not the culture conditions. The innate ability of the plant to multiply is very, very important. That is why sometimes you may get success very fast. Sometimes you need to wait for long, or sometimes you have to struggle hard to get the response. So the basic principle in tissue culture is balancing the auxin-cytokinin ratio. So always the inverse relationship is the very important. More auxin and low cytokinin. If that is the case, initiation of roots on cuttings of explants you can initiate. If you are moving towards lower side, so the medium range callus initiation will be there. That means auxin concentration will be moderate and cytokinin concentration will be less. Then callus initiation will be there. Suppose if the auxin concentration is very low and cytokinin concentration is very high, then it leads to the organogenesis, especially shoot formation, shoot bud primordia multiplication, lateral shoots production, like that. Since the shoot apex explant is very, very small, that is 100 to 500 microns in size with apical meristem 1-3 primordia, that itself is good enough to get a very good response in tissue culture. So the balancing the auxin cytokinin concentration at the beginning is very, very important, based on the explant nature and also what do you want to develop actually. That means whether you want to get the callus or whether you want to multiply the shoots. So, as you know, that there are four important methods of sterilization in plant tissue culture. Since you have to maintain absolutely what is it called, sterilized conditions or aseptic conditions, whatever tools you are employing in this plant tissue culture purpose, and you have to sterilize them properly. So sterilization by heat, that is the first step, where in the media and glassware we can autoclave it, or if it is a forceps and needles, and if you are using those are petri dishes, you can bake in a oven, in a hot air oven. And sterilization by filtration. Suppose certain hormones are heat sensitive, so before autoclaving the media, you are not supposed to add such compounds. So after autoclaving the media, if it is around 50 degrees or 60 degrees temperature, below 60 degrees temperature, before pouring into the culture vessel, you have to add through filter sterilization, nanopore filters or micropore sterilized filters. It's like millipore filters or whatever may be. Similarly, the air sterilization, the ambience, that means the culture room also should be free from the contamination of the bacteria or fungal spores, especially the fungal spores like cladosporium, which are highly predominant in the storage conditions. Storage controlled. Closed conditions. So in such cases, the air sterilization is very vital. Just like our laminar flow, here also the room curtains or door curtains we call mechanically. So they are fit with the HEPA filters, and sterilized only air only will be pumped inside. The most important thing is the explant sterilization. That means whether it is a leaf explant or shoot explant or root explant or seed or cotton leaf, the explant should be disinfected with chemically by either using a mercury chloride or calcium hypochlorite, like that is called commercial bleach, or hydrogen peroxide or 70% ethanol. So these are all the common things. So I don't want to go in depth. So what after adopting all these sterilization conditions, when your explant is you prepare properly, still there are various factors that influence the response from the explant. The first and foremost important thing is genotype and physiological state of the your plant material. For example, you are taking from the mother plant. Suppose the next time the same for the same, by using the same protocol, if you can get the material from the same genus and species from a different plant out, which is growing in a different geographical condition, there are chances of having micro variations, both physiological and genetically. So that leads to a varied response. Similarly, the culture medium, whether you are using macro micronutrients properly or not with the growth regulators or any other media ingredients, even slight variation, that also matters. And culture conditions and environment, the light intensity, the temperature, the humidity, all these matters. And also plant growth regulators like oxygen, cytokinin, gibberellins, or any other promoting agents. These are all the factors that decide the response. There are various constraints from tissue culture methodology aspect. First and foremost is microbial contamination that we can address, as I just mentioned, by using the chemical sterilants and also sterilization of the media and maintaining the aseptic conditions. Next one is explant establishment and the microplant of propagation due to the production of toxic phenolic compounds. Sometimes when you cut the explant, it oozes out various chemicals like phenolics. When you get exposed to the air, immediately the tissue becomes brown and die. So you have to use some like antioxidants like citric acid, ascorbic acid, which activated charcoal treatment, such things to protect the explant nature. Another important thing is the high cost associated with the chemicals what we use in the media, and the microplants derived from tissue culture are not fully photosynthetically active. So based on the photo period, whether it is 16 hours light, 8 hours dark, or complete light, the photosynthetic responses will vary. That also matters. And the gelling agent, when you are making solidified media, generally we use agar agar. Sometimes the phytogel you can use. So based on the quality of this material, based on the presence of the certain what is called lead or any other contaminants, this also matters. Impurities, any impurities that matters with the response. Generally, compared to the agar, phytogel will give sense very better response, because the impurities will be less. And the transpiration rate, that means when the explant is responding on a culture medium, you can see after 4 or 5 days the explant slightly curls up. or that means slightly fold. That means it is responding very fast. And that's based on the nature. Sometimes the transpiration rate also will be very high. That leads to, because of the transpiration, a lot of moisture gets accumulated in the culture vessel, and if the moisture content is more, contamination chances will be very high. Based on this transpiration, there is one technical term, hyperhydrosity is defined. This is the most important response in the tissue culture. Many people ignore, and I say this is very important because the hyperhydrosity in tissue culture methods can be dealt with appropriate media ingredients, appropriate exposure to the photoperiodic conditions. What is this hyperhydrosity? It is an undesirable physiological and morphological disorder. What happened here, you can see in this tissue culture bottle. Here, well, the left side ones are a little bit good and the new leaf formation organogenesis is happening. Right side of this experiment, you can see they are so fragile, crispy, and the shape is also a little bit distorted. When you touch such experiments with a forceps, you can see the cracking sound are very crispy, like snacks, fried snacks type. This is called hyperhydric conditions. This is mainly because of the these such a plants exhibit various biochemical characteristics such as reduction in dry weight and certain biochemical compounds present in plant cells like lignin, cellulose, chlorophyll. So you need to avoid these hyperhydric conditions. Otherwise, the people may overlook this one. The response in this so and so medium is very poor, like that. It's mainly because of various reasons, like the relative humidity inside the culture vessel, whether it is a petri dish or bottle, or ethylene accumulation. Whenever the response starts, generally plant tissues release the ethylene. The ethylene, actually, it is a senescence hormone. Once it accumulates more in the culture vessel, it shows the inhibition of further response from the explants. And also it leads to the hyperhydric conditions. The type and the concentration of the gelling agents, what we use, the absent cytokinin, especially the cytokinins and other ingredients. So how do you tackle this one? One is ventilation in the culture vessel, light intensity, temperature. So in earlier case, generally when we practice in laboratory, first and foremost thing is you will be asked to learn the technique of making a cotton bunk for your test tube or culture vessel. It should be so hard, because why we use cotton bunk is the exchange of airs to the micropores will be the cotton fibers will be pretty good. So that's why the accumulation of moisture inside will be minimized. But nowadays we are having plastic caps or screw cap tubes based on the vessels. Because of this reason, sometimes we will apply parafilm to avoid the contamination. So it creates accumulation of the gases inside. Gases exchange will be very poor, and it leads to the inhibition of the growth and hyperhydric conditions. So these are all various type of petri dishes or PS polystyrene disposable things or square shape based on the requirement. Even the Scott bottles also will be used for this one. So they, based on the nature of the plant explant or nature of the plant, you have to choose this one. So trial and error method we can use. We have to use these vessels. This is the latest technology. Some companies have developed. You can see just like an injection vial type thing. So before taking the injection, what we'll do, we'll just scrap the injection vial at that top and suck that injection chemical into the syringe and inject. Same thing here. The total capacity is just 2 to 3 ml medium only. If you are having a micro shoot, rather than wasting 50 ml media in a culture vessel, here you can use very little quantity, 5 ml, and you will save a lot of cost. It will save, and you can get the response also very good, because the complete surface area exposure to the photoperiod, that means 16 to 8 hours. In case if you are keeping, will be good, and it is just 130 mm in length. So such a type of polystyrene disposable things are available, and of course in India it's yet to get a popularity, so you have to import these things. Similarly, just like we are now discussing about this cross ventilation, that means exchange of the air, that leads to hyperhydrosity because of the accumulation of a lot of moisture inside the plant. So nowadays there are filters containing lids are available, because these adjustable filters are 0 to 20 mm in size, and in more labeling, filter placement to prevent the unwanted air leakage. For example, here one petri dish, one plastic culture vessel is there. Here one small groove will be there. Inside this, these filters, either one or two, based on your requirement, you can put and seal it. So gas exchange will takes place in this one without any kind. So it will filter all microbes, including fungal and bacterial spores, including virus particles also. The standard size is 0.22 nanometers size with a different sizes. So the diameter of this one based on the culture vessel. So hydrophobic material to protect the functionality of the filter also is available. So these are the great latest innovations in the culture vessels. So these things are also in abroad they are using, but in India it had to get on. Maybe cost is the one important matter. So these are the regarding the practical things what we face in the while handling the plant tissue culture. So there are various pathways of tissue culture. That is, one is the micro propagation. From a single explant, you can produce the multiple shoots. So that is a regeneration and organogenesis. It may be the direct regenesis from the explant like leaf or hypocotyl or root, or indirect organogenesis through callus. First callus will be there, then from callus it will come. There is another method called somatic embryogenesis. This is called, for example, if you put an inoculate, direct somatic embryos will be produced, or callus mediated somatic embryogenesis. And lastly, the synthetic seeds, which are either coated or non-coated, desiccated somatic embryos. So these somatic embryos, for example, in a normal petri dish, you can maintain 3 months, maximum 4 months. Then they turn to brown, or they may regenerate into plantlets. But if you want to keep intact these somatic embryo for future use without further rapid growth, you can embed it into some alginates, that is called encapsulation methods. So you can protect, and whenever you want, you can use these things. So these are all the 3 important things to get the true to take plants in plant tissue culture. This is just a picture I have taken from my old records wherein we worked. This is a plant called Anoto. So here from the one X plant, you can get the multiple shoots. You can say 1, 2, 3, 4, 5 like this. See, similarly, this is the Cotlander leaf X plant. You can get multiple shoots. Here, this is also here. See the petiole still attached to the base of the leaf X plant. From there also you can get the direct shoots. This is the nodal explant of vanilla plant. You can see at least a bulb, at least 10 to 15 multiple shoots you can get from one axial bud. This is again a hypocotyl portion of a cut explant at the collar region where root joins. That is dissected, and from that end you will get this multiple shoots. So this we have very well optimized for coffee plants and also annatto plants. Similarly, anther culture, and even in a reactors in a closed vessel, you can get a high number of minimum 40 to 100 explants, 100 microshoots. This is a stevia in a reactor. So just now I'm mentioning the couple of slides before I'm mentioning about the importance of the light. Generally, the most commonly we go for 16 hours light, 8 hours dark. Here, generally we use incandescent bulbs in earlier days. If it is algal cultures, normal golden yellow color lights are preferred. But for general tissue culture, fluorescent tube lights we use. Now it has been replaced with the LED, that means light emitting diodes one. So you have to optimize the wavelength of this one, and you can use these for very effective multiplication, whether it is for a callus multiplication, whether it is for the roots or micropropagation, or even for producing the secondary metabolites. That point so we can discuss in the subsequent slides. Since our topic of the plant tissue culture is mainly focusing on the food, so here I would like to mention how this plant tissue culture is helpful to improving the food security in India. So first we should know what is food security. The name itself is giving. That means you have to provide some assurance of getting the food for the needy population in in the near future. So food security is the capability to assure on a long-term basis that the system provides the total population's access to a timely, consistent, and nutritionally adequate supply of foods. It's not getting junk foods and other things. If somebody donates eating like that, not like that. Nutritious food towards the malnutrition-related disorders, that is also very important. That also comes under the food security, like pulses, cereals, fruits, vegetables, and milk. From tissue culture point of view, even though this very important high-end, high-yielding varieties were developed, especially for the wheat, ragi, and sorghum, like any other plant. Of course, rice, but in rice is most commonly cultivated through the breeding techniques. Similarly, tissue culture of fruit varieties like mango, guava, banana, papaya, watermelon, apple, these are the most successful among the fruit varieties. And micropropagation of vegetables like potato, carrot, tomato, brinjal, these are n number of plants have been taken up. But these micropropagation among these potato, carrot, tomato, brinjal occupy the major role, major importance. And the tissue culture of nuts, cashew nut, groundnut are the predominant ones. So here I would like to touch a plant tissue culture of green leafy vegetables and food value plants. Why do we need green leafy vegetables? There are n number of green leafy vegetables are there in India. Again, it varies with the cultural diversity from one state to another state. Sometimes things are common, certain things vary. Even in abroad, vegetables like lettuce, kale, some variety of vegetables were there, leafy vegetables. So in India, equally spinach, amaranthus, other varieties were available. Nowadays, many companies, even in India and abroad, they are growing these leafy vegetables in a controlled conditions in a poly houses, wherein the LED lights will be fixed because under specific LED light, the growth responses are pretty high and very fast and luxurious growth will be obtained, and also the nutrient contents also will be enhanced. So this is a red, yellow, blue, green light. Such a LED you can adjust for this one. In our CFTRA also, we have developed very important protocols, micropropagation and tissue culture protocols for the Rumix basic areas. This is a local delicacy again in the southern states. Sorrel, blad and sorrel plant, they call. It's very sour in taste and very tasteful and very good content of iron and other organic, especially organic acids content is pretty high in this one. So we have developed a very efficient tissue culture process technology for this one. This is another plant called Malabar Spinach or Bacilla rubra. You might have seen in our places also. So basically it is a backyard plant. Two different varieties are there. The green one is the Bacilla alba. The red one is the Bacilla rubra. Since we are working on natural pigment for since long time, we want to explore beta lines potential from this plant. As a part of this course, we have developed first time tissue culture process for technology for this Bacilla rubra. You can get very efficient clones and other things, and you can easily harden these things because this is a partly succulent plant also. This is another plant called hibiscus sabdariffa roselle. There are two different varieties of hibiscus edible ones, that is hibiscus cannabinus and hibiscus sabdariffa. Both are the most sought after green leafy vegetable in the state like Andhra Pradesh and Telangana and Tamil Nadu, of course in parts of Karnataka also. So this big, since it is a very good source of again iron content, organic acid, ascorbic acid and other essential minerals. We are working on this one, especially dehydrated foliage based food formulations for valuation. As a part of this, again, we have developed an efficient tissue culture process for this green leafy vegetable. And this is a picture where you can see this annatto plant. This is the annatto dye. That means it is one of the important apocarotenoid. Even at more than 100 degrees temperature also, the dye is highly stable, unlike other carotenoids which lose their pigmentation capacity index above 50 degrees temperature. So the various varieties are there. For different varieties, we have developed a very efficient micropropagation method and a very high important somatic embryogenesis and genetic transformation methods for this one. And we could able to improve the annatto pigment from 2% to 4% through our tissue culture technology and also through elicitor mediated technology. So at this point, I would like to touch a few slides about the secondary metabolites. Without secondary metabolites, the tissue culture discussion will not be completed, because it is not alone getting the micropropagated shoots or mass multiplied shoots through tissue culture. Even the secondary metabolites play an important role in this tissue culture, because these are all secondary metabolites, means they generally use for the plant for the defensive purpose and won't involve either in photosynthesis and respiration. So byproducts of photosynthesis and respiration, these secondary metabolites will be produced. They are pharmaceutical important, medicinally important, and food value industrial important metabolites like that belongs to various functional groups like aliphatic, polyketed, benzofuran, like that n number of chemicals are produced. One best example I want to give you is vincristin vinblastin from this periwinkle, the Vinca rosea. This is the most sought after highly expensive medicine extracted from these petals and also through tissue culture. That is commonly used for blood cancer, leukemia purpose. So the production of these secondary metabolites like plant tissue culture mainly depends upon the precursor feeding. That means a biosynthetic pathway precursor chemical you have to add to the culture medium and inoculate your callus, so it will be converted to the end product. Or influence of growth regulator, permeabilization, optimization of culture conditions, or certain biotic, abiotic stress you can create and improve the content, or selecting high yield varieties. I don't want to go to this one because it is a huge topic. Similarly, suspension cultures, instead of shoot cultures and going for hardening and taking tissue culture plant to the field, even the callus cultures you can multiply and produce a number of products. The advantages are here that constant regulations over the various stages of operation. So, for example, initially micro shoots, to shoot elongation, in vitro rooting, hardening. So all these stages you can avoid. Your time will be saved because rapid multiplication of cells will take place, and the cost you can bring down. Very easy to handle. Within a short time, you can produce the uptake of nutrients from the medium by this homogeneous mass of cells will be very high. Easier production and very high. And there are various advantages for using cell culture technology. This also in depth I don't want to go. So once you optimize the callus cultures, you can make the suspension cultures in the liquid medium without agar, and step by step you can scale up to the bioreactors. In bioreactors, in even 50 to 100 liters capacity bioreactors are available for producing the medicinal important secondary metabolites in abroad. Then you can, by using these bioreactors, you can produce these pharmaceutically or medicinally important metabolites for evaluation purpose. There are certain other important reactive metabolites like phytoestrogens. You know the estrogen hormone importance in female reproductive system in humans. This is very important because especially this estrogen hormones plays a very important role during the postmenopausal symptoms. If people use certain food materials wherein the estrogen related compounds that is called phytoestrogens are predominant, it alleviates the hot flashes related problem, postmenopausal problems, low bone density, osteoporosis related issues in women. So these isoflavones exactly mimic the structure of estrogen. That's why they are called phytoestrogens. One such important thing is soybean, where you can get genistein, genistein, biochemical formaldehyde. Unfortunately, these levels of these phytohormones, these phytoestrogens will be moderate in these cultures, but even though the beans, it is very high actually. So what through tissue culture technology, we could able to produce 3 to 4 folds more content of these phytoestrogens in the tissue culture tissues and tissue culture methods. That is the beauty of tissue culture. So we have developed suspension cultures for this one. At this point, I would like to mention one slide on the importance of the statistical tools. Six Sigma technology is nowadays people are using, or its other developed versions. Because in business world, generally, this Six Sigma technology will be used to deliver double the competitiveness among the employees and to get the more profits. Especially the DMAIC is the one important step commonly used in this Sigma technology. One is define, that means identifying a customer needs, what is required for how to address the problem. Then measures, you have to book certain measures by collecting the basic information and putting the benchmarks. And you have to logically, you have to analyze what is the problem and root cause. Next, how to improve that one, and how to do all these things in a controlled condition. So all these things matters a lot. So basically, based on this one, you can design the experiment. So DOE, design of experiment, for scale-up studies by using the statistical tools is very important, wherein ANOVA methods and other statistical tools are blended with this one. Suppose you are using 3 different combinations of hormones. Okay, your target is to see the multiple shoots or my root number, for example. So 3 different hormonal combinations, 2 different responses. So how many variants are there? How many factors are there? So based on this one, you have to design the experiment. There are 2 different methods. One is the Raymond Montgomery model, and another is the Montgomery's Box-Benken PBD model. The first one is a first order reaction. The second one obeys with the second order reaction. In the first order reaction, mostly the response surface will be the curvature relationship. That means between the factors and response, if you plot a graph, mostly the curves will be there. Based on how much, to what action the curve appears, so you can optimize the exact concentration. But nowadays, second order reaction based ones, that is called RSM technology, response surface methodology, wherein box Benkel reactions will be used, where it is more or less it's a non-curvature type things. So I don't want to go in depth again in this one. It's a bit complicated. So some engineering formulations also you have to apply. The same technique we have applied for the soybean in vitro cultures to optimize the exact concentration. Because suppose if a normal tissue culture, if you use 2,4-D some concentration, NAF some concentration, kinetin some concentration, you will get some response. But if you use this RSM method, the exact precisely how much concentration at is the optimized one that gives the maximum response of biomass and phytochemicals will be secondary metabolites you can optimize. That is the beautiful side, because even 1 milligram saving of the hormone matters a lot when you are going for scale-up studies. So like this, we have developed this tissue culture-based things for the soybean, and also the encapsulation methods by using potassium alginate, sodium alginate also for these callus cells we have done. And this also improves the soybean, this phytoestrogens content a lot. Of course, genetic transformation studies for improving the SOD activity, antioxidant-based activity, and also producing other omega-3 oils also we have done in soybean. It is not alone these secondary metabolites, even natural pigments you can produce in these cultures. This is the Bacilla rubra plant. The fruit of this plant contains the beta lines. Otherwise, for optimization of the conditions, whether it is possible to produce these beta lines in vitro conditions, for the first time we have developed. This is a leaf-based callus. Slowly you have to do 5-6 subcultures, then segregate the pink colored cells, then multiply those things, and you can get the beta lines in vitro production. This is one of the recent accomplishments from our side. Similarly, carotenoids also you can produce in root cultures. Very hardly will find any carotenoid pigment production in vitro in using the plant cultures. Here, this is the root cultures of the annatto plants. These are all normal root cultures, not hairy root cultures. There is no transformed roots. These are all normal root cultures we have multiplied, and if you can extract the pigment by using the solvents. And these are the hairy roots. These are the normal root cultures again from this hibiscus cannabinus plant. That is the plant I showed you sometime back, a few slides before. That is a green leafy vegetable. From the leaf explant, you can separate this one and multiply in large culture vessels. The beauty of this one is from these root cultures, very high content of ascorbic acid you can extract compared to the external plant grown plant leaf. 3 to 4 times more ascorbic acid content these roots are able to produce. When you are doing the tissue culture, the variation is very less. But sometimes, because of one or other reason, cultural practices or media and genetic instability, variations will happen in the somatic tissue. So that is called somoclonal variants. These somoclonal variants are whether they are reversible or not, that is a debate. Sometimes it is reversible, sometimes it is not reversible. If it is a genetic level, it is not reversible. Suppose if it is an epigenetic response, that means genetically they are stable, but because of the environmental conditions of what we apply at the tissue culture, the protein expression part varies. But such things, if you bring back the normal conditions, the epigenetic responses will be reversed. So these are called somoclonal variants. So without harming the plant, you can do. One model example is coffee. In coffee contains very important bioactives like chlorogenic acid, and cafestol, kaival, like trigonelin, etc. Especially the cafestol and kaival appears in the creamy. These act as an anti-nutritional improve it higher levels of cholesterol. So we want to use the somoclonal methodology to develop the coffee plants that produce very low content of this cafestol and kaival. So by using the somoclonal method without any genetic transformation, we could able to successfully develop somoclonal varieties of plants that drastically reduce 90% of the existing cafestol and kaival. Nowadays, the industry importance is very high in the tissue culture. Still in India, of course, we get labor at very cheap cost. But nowadays, because of the manpower cost increases, it is a bit costly. Otherwise, a lot of manpower is available. But in abroad, everything is robotized to minimize the cost. So robotization and automation in plant tissue culture can reduce the labor cost, which accounts for a high proportion of the profits. So here you can see it exactly choose. You can program the explant size and condition. So such explants automatically it will pick up from that one. It itself feeds the dissects the shoot tip explants or nodal explants. It will pick and also inoculate in the culture tubes. So by using this technology, even 15,000 plants you can handle within an hour. Just like when you are doing a high-end PCR, a strip of tubes of 10 to 15 will be there. So multi-channel pipette you can use and you add your reagents and finish the experiment for PCR. Similarly, strips of tubes with attached to each other will be there. So it is since it is an automated version, this is called a nick in system. It is a brand name. So the machine itself will pick that explant and automatically at a time it will inoculate, so that you can save the cost, time a lot. But of course, initial expenditure will be very high unless if you do an industrial scale. It is not much profitable for the lab based studies. So ultimately, the target of this tissue culture is of food is to take the plants of food value from lab to the field. So initially, we will grow the tissues inside, multiply, these harden and produce. This is a typical example of one banana variety. It is the Nanjangud Rasamalai variety. Delicacy, local delicacy to the Mysore region. This grows only in the soils of Mysore region, that is called Nanjangud. That's why it had a GI tag. If you take and plant in any other location, it will not grow. One small fruit of this cost is around 20 to 25 rupees. Similarly, there are other varieties we have developed. This is a red banana variety of near Kerala. So for this also, CFTI has developed a technology. It is a very rich source of alpha and beta carotene, up to 600 micrograms per 100 gram of fruit. One fruit will be approximately 250 grams to 400 grams. You imagine how much beta carotene will be there. So for that also, CFTI has developed tissue culture technology. So ultimately, this once you optimize this technology, if some entrepreneurs buy the technology and they can on their own they can establish this in a micro startup level. So this customized cultivation is nowadays very common for many agricultural crops. Similarly, tissue culture plants also in a customized way people are now doing. So at this point, even though my intention is not go in depth to the plant transformation technology, but it is very essential to touch here a couple of slides, because the plant transformation technology is helpful to transfer the genes of desired from one plant to the another one, or to insert a desired gene into a targeted plant. This is mainly through the various methods like normal agrobacterium mediated transformation methods, or by particle bombardment, or by using the chemical peg methods, wherein the bacterial plasmid will be mobilized through into the plant tissue and integrate with the plant DNA, and it will multiply, mainly because of the various specific genes specific to this plant that is called Vir genes. So it could be either agrobacterium tumefaciens mediated transformation or agrobacterium rhizogenes based transformation. While doing the transformation, to easily identify the transformed culture, either GUS gene will be inserted like a marker gene to identify, or GFP gene will be, fluorescence protein gene will be there. So whichever culture gives this fluorescence or this blue color upon staining, that gives indicates the presence of this culture is transformed. Then by molecularly, by using this with genes or tDNA or location, or by role genes in case of agrobacterium rhizogenes, so you can confirm that it is a transgenic plant. So this is called agrobacterium rhizogenes mediator. Here one explant is there. You can see initially hairy roots are inserted. So the beauty of this hairy root is once you detach and again inoculate in the fresh liquid medium, it will multiply. So uncontrolled growth will be there. This is called hairy root technology. This technology has revolutionized, especially to produce the various secondary metabolites of importance, whether it is a food value or pharma-related things in industry. So again, this hairy technology is also useful for molecular breeding, phytoremediation purpose, and also to understand the plant biosynthetic pathways, root physiology like this. So various purposes, especially the phytochemicals, as I mentioned. And even you can produce the various color pigments like anthocyanins. So see, here it's the green leaf. The anthocyanin biosynthetic genes are inserted by the scientists into this one. So the plant what you get contains a completely of the pigment, and that pigment, the left plant you can culture in vitro based on the media constituents and the level of gene expression. The pigment color will vary. Another important contribution wherein we were working for the past 2 decades is on coffee. So the coffee, as you know, the caffeine is the most important thing. To some extent, it's okay, 1 or 2 cups. If you drink too many cups, it leads to the adverse effects. So the decaffeination coffee products are 30% occupancy in the market. That means 70% of the market is for the caffeinated coffee or caffeinated coffee based products, or 30% is for the decaffeination. Generally, solvent based methods are used, but there is a health hazards will be there for the solvent based remnants. So for that, tissue culture through agrobacterial transformation technology we adopted, wherein we want to produce the decaffeination plants. The most important thing, theobromine to caffeine, that is by N-methyl transferase. Third one, the second N-methyl, that is called theobromine synthesis. It produces from the 7-methyl generator, this one. So by using the antisense, initially we have developed very efficient technologies for the coffee variants in India, both Arabica and Coffea canifera, and also optimize the transformation technologies. By using the antisense and RNA technology, we developed coffee plants wherein 99% of caffeine has been completely down-regulated in this one. And even for Arabica plants, we have developed the transformation transgenic technology. Here you can see the very beautiful expression of the gus in the somatic embryos and also the transgenic plant leaves. So there is another method called somatic hybridization. Of course, we have not much stepped into this one, especially for the ornamental and floriculture crops. This technology is very well adopted. It is called somatic hybridization. That means two somatic cells gets hybridized. For example, this is the cells isolated. First, cell wall and other things gets be dissolved, and the protoplast will be extracted, and the fusion of these protoplasts takes place to the integration of genetic material, and new varieties will be there. This is also very commonly used, especially in the crop improvement. As I mentioned in the beginning slide, this artificial seed or synthetic seeds also you can develop, wherein you can preserve the germplasm for many, many years also. So in between every 1 year or like that, you can redo this same method. So the multiplication of a non-seeding protein plants, hybrids, or propagation of ploidy plants, this is very important. And germplasm conservation is the main thing. Here you can see the that shoot tip explant or like a leaf explant has been embedded in the potassium alginate cells, and these will last for 5 to 6 months based on the concentration. Whenever you want to, you can again put it in a solution. So potassium alginate will get dissolved. Your explant, you can put it in a medium and you will get a plant. So this technology is called synthetic seeds. If I don't touch this topic like biofortification, the today's topic will not having a justice. So why we means biofortification is to improve the nutrition status. For example, rice, you can see the raw rice contains, for example, 8.8 ppm of iron. When you completely go for completely polishing, 50% gets diminished. So like this, many vitamins and micronutrients gets washed away during this polishing of rice. Because of eating this polishing rice and other low nutrient content items, people are becoming malnutrition, facing the malnutrition problem with low iron and zinc. For because of this, the golden rice has technology has emerged, wherein the carotenoid producing genes were integrated to the plants, especially the rice plant, that is called golden rice. By using this technology, scientists could able to improve 4.4 folds of more carotenoid in the rice plant along with the zinc plant, because both iron and zinc association will be very good. Homeostasis will be very important. Both together, if you insert, take through the food chain to the human body, their bioavailability will increase a lot. So that's why by using this technology, this transgenic golden rice, a lot of carotenoid improvement has been achieved, and this could address malnutrition thing. In many countries like Malaysia and South American countries also, it is very commonly used, but not in India. Similarly, other biofortified crops like banana is there, maize is there. Similarly, some millets are there. Tuberous crops like sweet potato and cassava are there. For example, this is the cassava. This is normal cassava is if the entire starch portion is white, but we have vitamin A enriched one. The transgenic technology is there. That is another one. Similarly, in sweet potato, normal variety, white variety is there, pink variety is there. Again, in the pink variety, wherein lot quantity of anthocyanins are produced, in this one, IBOR in gene has been overexpressed by the researchers, and at least the sweet potato variety with the more carotenoid content. And because this sweet potato and cassava are the most staple crops and food materials in the African countries, a lot of malnutrition related problems were addressed by using this one. Similarly, now calcium biofortified transgenic plants are also available. Why I'm telling these things is for all to achieve all these things, your tissue culture is the basic tool. Without that, you cannot achieve this one. So Park et al. In 2009, decade back, have generated transgenic tomato, potato, lettuce, green leafy vegetables like lettuce and expression, wherein calcium transporter genes they have mobilized. So they give lot of more, accumulate more calcium. So without using the extraneous calcium and vitamin D based tablets, these crops acts like a staple generators of the calcium for the human consumption. Similarly, the golden bananas are also available, developed wherein phytoestrogen synthesis gene that involved in the carotenoid biosynthetic pathway with ubiquitin promoter has been inserted. This is the green banana. This is the banana you can see here. It's a normal banana. So beta carotene levels based on the intensity of the gene expression it will be there. So by using this one, consumption of 300 grams of transgenic banana, it provides 50% of the vitamin A that required for us per day. So suppose if you require a vitamin A, some 95 units of international units of vitamin A, 45 units you will get by using this one banana itself. So the question is now whether we can adopt these tissue culture technologies for food crops is or not. It is already there, especially it is very, very successful in abroad, and also from pharmacy aspect, for a secondary metabolites point of view, it is very, very important technology. But whether it is the food crops or for non-food crops or commercial ornamental crops, the cost is very, very important. So, due to the higher costs, a cost benefit analysis of in vitro culture is wise before implementation of the technique. So, you have to initially plan for the mass multiplication and mass propagation from lab to greenhouses and then to the fields. If your the size of your target is low, cost always will be very high. And also based on the requirement, you have to adopt these industrial the latest developments like what I showed, culture vessels or automatic robots and other things. Then it will then in abroad it is very, very successful, but in India of course still we are relying on the most labor intensive methods only. To achieve this one, certainly the novel technologies like gene editing technology and environmental factor manipulation are very, very important. All so far we have now discussed many things about the antisense technology, RNA, like that you might have studied. But nowadays CRISPR-Cas9 technology, the CRISPR-Cas9 technology, it is a gene editing technology. It is not actually exactly any transformation technology. From a particular thing, one particular nucleotide change will be that you will remove 2 to 3 bases you will remove it. It maximum 10 to 12 length. This is called gene editing technology. Just we are editing. So this technology has revolutionized the crop plants. So again, there also again tissue culture is a tool. Similarly, environmental factors play an important. Stress is the most important area. If you hit Google in tissue culture or anything, 50% of the publications related to the studies related to the stress, whether it is a biotic stress or abiotic stress, initially at the in vitro condition, then during the hardening condition, then at the field conditions. because with little change in the stress conditions like biotic or abiotic, a cascade of genetic expressions will be varied. That is called differential expression of genes under stress condition. That alters the protein structure, that alters the secondary metabolism, that alters the morphological features. So by using this hereditary mediated technology only, we also developed improved the anatomic pigment to 4% that meets the international standards, come 2% of Indian levels. Earlier, we are by using the elicitor mediated technology at milligrams or micrograms, you have to use the elicitor. But now nanotechnology also entered to this tissue culture, where instead of micrograms, so you will use in nanograms the elicitor. That is called nano elicitors development, wherein metal or silver, strontium based tagged elicitors also will be used. So for all these things, this tissue culture technology will be very useful. So you can improve the leafy vegetables, you can improve the food value plant, you can improve food value metabolites like caffeine, capsaicin, etc. Similarly, isoflavones and other important secondary metabolites by using this technology. So if you are any students who are interested in this one, so these are some beginning topics in tissue culture. Just I want to give a glimpse. I don't want to read this one. So how vitamins affect in vitro organogenesis in tissue culture, how to develop plants from the cotyledonary tissues, how to develop organic extracts in tissue culture, how to generate the large biomass for genetic transformation. Similarly, how to use probiotic carbohydrate for inhibiting lectin adherence, and also plantlet regeneration, in vitro responses to shoot organogenesis, like this various similar transgenic technology in this, how plant growth regulators like oxygen, cytokines, gibberellins, and even triacontanol and other chemicals will influence. Like this, even the carbohydrates, generally sucrose we will use in the medium. Suppose if you use glucose, fructose, or any other carbohydrate sources, how the response will vary, especially in some plants which are recalcitrant. If you don't get somatic comprehension, so you have to replace sucrose with some other carbohydrate sugar source. So like that, you have to optimize. It's a trial and error based method if it is a new system. Otherwise, it is a well-established system. It is okay. But my experience says that even if it is very well established, for example, in coffee, I have done in coffee a can for a lot for 5 to 6 years. For all Indian germplasm, we have developed a tissue culture technology. When I had been to the France for an institute of coffee genomics, wherein I worked for coffee genomics, initially they asked me, since you are expert, why don't you develop a tissue culture technology? What for our plant system? It took for me 6 months to get a response out of 100 to 100 different sets of readings. I mean parameters. Ultimately, only one plate I could able to get response in that particular variety. So even the genetic both belongs to coffee, a can for a robusta coffee, but the variety matters. From one plate, in the next 3 months, I could able to elaborate 400 plates of somatic embryos that I used for the transformation. So you need to master over this technique. You have to learn this one with a very inquisitive observation tissue culture. Otherwise, 70% of the responses you will overlook, and you will lose without noticing itself. So persistence, patience is very important in plant tissue culture. These are all different companies globally involved, key market players in plant tissue cultures in abroad, whether it is Europe or Western side, Alpha Laboratory, Dark Necessary, DP High Media Laboratories in India, of course, we might say Sigma, also there, Nighthall, Labland, Dachafa, many companies are there, plant media. Thank you all for your patience listening. I hope I have justified my talk with this one. Hello?
Yes, sir.
Yeah, thank you.
Sir, just give us 1 second.
Yeah.
Participants, you can ask your questions in the chat box. Our speakers will address it.
Can you hear me?
Yes, yes, Redisa, we can hear you.
Good morning, sir. How are you,
Good
sir?
morning.
This is Dr. Jairam Reddy,
Fine.
sir.
Yeah,
It
yeah,
is,
yeah.
it
After
is
a long time. Thank you.
after a long time, sir. So it
Yes,
was a
yes.
very good and highly informative talk, and you spoke very well about plant tissue culture and especially on the industrialization and robotization of plant tissue culture, sir. I have a basic question because you're
Yeah.
working in the industry. Um, how far is it practical, the application of plant tissue culture in the development of secondary metabolites, practically or industrially? Can you tell me about that, sir? Because I don't have the knowledge about the practical success of plant tissue culture in terms of producing secondary metabolites by using, let's say, bioreactors and things like that, sir.
Yes, sir. Actually, I didn't put much information because of the time constraint into this one. Some time back, I had given a talk exclusively on this topic.
Yes.
How to what extent we can get a success in this one, especially in the Germany and Japan and one more country. Yes, Sweden and Netherlands. These are the four countries wherein this tissue culture based secondary metabolites production has been taken to next level, because a lot of chemicals, industrially important chemicals are produced by using this one. I forgot that particular one name that is used commonly in a anti-cancer purpose. That is Japan is producing, even till today they produce that one. So the cost will be very very less. Many people are not having, even in textbooks also you don't find this information. So I have collected from various sources and I have depicted. So certainly there is a great boom for that, especially the buying the bioreactors. So even 20 to 30,000 liters of bioreactors in Germany and other countries, they are running for producing the secondary metabolites. It is there. It is certainly there.
But how far is it practical in India, sir? Because there are a lot of
In,
potential. For example, I know one of my students, Sujata, you remember, she worked on saffron in your lab. She did a PhD.
yes.
You remember, sir? I came and visited, and I also was there for some
Yeah,
time.
probably.
She worked.
Yeah, at the time, Dr. Ravi Shankar was there.
Yes, yes, yes.
He is the
I
pioneer in
worked
the
in
saffron
CFTRF.
research.
Yes, I worked in CFTRF for 1 year, sir, with Ravi Shankar. So
Oh, good.
that time Sujata was working on saffron. I remember I saw the cultures
Hmm.
also. The media was having saffron in color. The saffron was induced in the media, but I don't know what is the practical application of that, because you know very well saffron is so expensive, and producing it in the wild and in the agriculture field is very difficult. But if you can develop some method where you can produce saffron directly in the tissue culture medium, and I want to know what is the practical success of this, sir.
Yes, sir. Especially with the saffron in the RRL Jammu. Now, this is also CSR laboratory in the Jammu. So they are also pioneer in this saffron-based work. Of course, Dr. Ravi Shankar in CFTR has done a lot of work on tissue culture and biotransformation work and somatic embryogenesis. But RRL Jammu has gone even for the heavy root technology and suspension based culture technology. They have developed, they could able to go to certain level, I mean 1 liter or 1.5 2 liters level only. Beyond that, I don't know the reason why they didn't scale up this saffron in production. But certainly one thing is, if there is a difficulty in understanding or elucidating the pathway, biosynthetic pathway of any secondary metabolite by using the native plant system, this tissue culture technology also can be used as a model. But certainly, as you have mentioned, this saffron in production under in vitro conditions has limitations. Maybe bioreactor capacity, or I don't know. So, but RRL Jammu has done up to 1 liter, 1.5 liters capacity also. Even Spanish groups also did on this one.
Yes. Thank you so much, sir.
Yeah.
Over to Tanvi. Tanvi, you can take the other questions from the participants.
Yes, sir. Thank you. So we have a question from Sridevi ma'am. She is asking if sir could elaborate a bit on Sigma 6.
Yes, this is the software actually in industry, as I have mentioned, to improve the competitiveness and the performance. So, if you competitiveness, if you apply to the in vitro technologies, I relate this to the media compositions like growth regulators, and performance means the response what we get for a any desired for example multiplication biomass or secondary metabolites. So for example in a company, so 10 15 employees are there. So you can make them into the three different groups based on their knowledge or based on their IQ or based on their maybe work load or work performance or like that. And ultimately you should get a response. Response is one means productivity. Productivity in terms of material development or software development or whatever maybe. So what happened is here three different sets of groups are there. Here two different responses you are anticipating. Between these two, you have to optimize. That means if I provide a basic incentive to the employee, for example, 100 rupees, whether I have to give 100 rupees incentive to the developer to give to the employee to get maximum output, or I have to give only 50 rupees or like that. So it is very difficult in a human tendency to understand. Similarly, if you apply the same things to the in vitro cultures also, for example, 2 grams of 2,4-D, 0.5 grams of kinetin is required to get a biomass of 1 gram or 1 milligram of a chemical or a secondary metabolite. Suppose that is what you optimize in a culture vessel. Suppose you are going for a 1,000 liters reactor. In 1,000 liter reactor also, per liter means 2 grams of 2,4-D means you have to use 1,000 into 2, 2,000 grams of 2,4-D and 1,000 grams of kinetin. It is very, very expensive, but it may not be exactly required. In a small flask, when you are doing, the condition is different. When you are doing in a reactor, the condition is different. There, instead of 2 grams, so if you are able to manage, get the same response for 1.5 grams of this one, 500 grams of 2,4-D you will save, and you get the same response in the bioreactor. So to exactly precisely know the exact concentration of the growth regulator for a desired to obtain the desired response, these statistical tools are very important. By loading this ANOVA and all other factorial designs are loaded in a software, and ultimately they developed that is called RSM technology, the response surface methodology technology. So it is the response surface methodology is a second order reaction obeyed response. It is a part of methodology that evolved from the Sigma. But unlike Sigma software, where black belt, green belt, like the different belts they award for the industry-based people for learning these skills. But in tissue culture, actually, it is not there. This technology is very commonly RSM technology useful in a crop in crop improvements, agricultural biotechnology, and also in the tissue culture technology, even in the food engineering also.
Okay, thank you, sir. Next question is from Pushkar sir. What are the
Yeah.
challenges faced when plant cell suspensions are used for production of secondary metabolites?
Yeah, the first thing is the subculturing. For example, chili plant is there, capsicum. There you can get callusing from the quaternary leaf or hypocotyl explant within 10 days initiation. By next 2 to 3 weeks, you will get sufficient biomass. But the callus is very hard, light green, and hard also. But such a type of callus is not useful for making the suspension cultures and also to get secondary metabolites. They very fast they turn to brown. So little quantity you have to take, and the same medium you have to subculture. Every 3 weeks to 4 weeks, minimum 5 to 6 subcultures are required. Because of the frequent subculture, it adapts to the in vitro conditions of the medium, and it will become very what is called crisp, powdery, and also light yellow color callus will be produced. Such a callus is very good for the further multiplication. Such a callus after 5 to 6 cultures if you calculate the biomass, rather than 1 to 2 grams of biomass in first 15 days, after 5 subcultures you will get 10 grams of cal. That means 10 times more biomass it will produce. Similarly, such a callus, if you put in a liquid medium, you will get a uniform suspension. Otherwise, the clumping will be there, like clumping. The clumping, why it happens means when a more ethylene circulation inside is there, or proper shaking is not there, or media ingredients, more cytokine is there. These are the problems. So such clumping will be one problem in suspension culture that you can avoid by optimizing the conditions. 120 rpm on shaker you need to put initially 70, then you can go 100, and the next subculture 120 rpm. Once it is in 120 rpm, there will not be any clump formation. But once you get this clear suspension, you can easily handle this one. Biomass you can separate by using Wattsman filter paper, and metabolites you can extract by using the solvent based methods.
Okay, thank you, sir. I think that was all that we had for question and answer session. So we will proceed with the. Okay, just a second. Yes, there is one more question by Devi ma'am. What are the characteristics or signs of a good callus, and how do you differentiate them?
Yes, good callus means you should when the multiplication is happening, you can see the visually also you can see the healthy nature of the callus. Sometimes what happens is generally for a good callus, I say by 14 days to 21 days, that means the second week ending to the third week, maximum proliferation of the callus biomass will happen. Very rarely fourth week also it will happen. Afterwards, the curve will be declining. That means rather the biomass gets stabilized or it won't multiply. When a hormones like 2,4-D are there, you must be very careful by not allowing the callus to grow on the same medium more than 1 month. Otherwise, the phenolic substances will be produced and it turns the callus cells into brown color, and cells also may die. Sometimes you can see on the bottom side of the petri dish or culture vessel, black color formation or color dark brownish color formation. It is just because of the leaching of the callus, these phenolic compounds and other things. So you have to take immediately the green superficial callus and subculture. This is one important thing. Similarly, too much hardness should not be there. Fragile nature is there. Fragile nature of the callus is very important. That is the one indication of the healthy callus. Unless you get the fragile nature, neither you get organogenesis, nor you get somatic embryogenesis, nor you get good callus biomass or metabolite production. So that fragile callus is very important. You can see under a microscope with a little staining the healthy nature of the cells also. So that morphology you can see under a microscope also the nature of the cell.
Okay, so we will move ahead with the session as we don't have any more questions. Thank you, Giridhar sir, for such a wonderful session.
Thank
Now
you
I
very
would
much,
like to
Shivala.
request Mr. Hitesh Gura to introduce our next speaker. Over to you, Hitesh sir.
Thank you, Tanvi ma'am. Good morning, all. It's my great honor and pleasure to introduce the speaker for the second session of today's Biofacet, Dr. Jairam Redisa. He is a professor and the head at St. Joseph's College, Bengaluru. He had conducted research at IIHR and IISc Bengaluru for his PhD. Sir received his PhD in the field of biotechnology of orchid in 2002 from University of the Bangalore. He guided 4 PhD students so far and currently guiding another 2. He had collaborated with the Wageningen University, Netherlands, for the research in gene cloning and bioinformatics. So far, he had published more than 92 research papers in renowned scientific journal and authored 9 books. He had revealed and deposited structure of the cancer-related protein MAGE-E1 in Protein Data Bank. In 2015, National Environmental Science Academy honored him with the Eminent Scientist Award 2015. St. Joseph College, Bangalore presented him a Scroll of Honor Award for his scientific achievement in 2016. He is a recipient of G.R. Patel Dronacharya Award for Academic Excellence and J. Agarwal Gold Medalist Award from the International Society of the Gene and Cell Therapy. He uh, also awarded by the Puraskar Award in 2021. It's our pleasure to have you here, sir, for enlightening all of us by your knowledge. Thank you very much, sir, and over to you, sir.
Hello, Jairam, sir. Hello, Jairam, sir.
Hello, good morning.
Yes, sir. Good morning, sir.
I would like to share the screen.
Yes, sir. You'll have to
Okay,
click the
I
button
should.
to
I've
share.
pressed
To
the
share.
share button. Share
Yes, sir.
button.
Correct.
All right, and there are three options: slides, video file, and share screen. Which one I should select?
So share screen.
Share screen. Okay, I
Yes, sir.
click this share screen, and
Yes, sir.
what should I mention now? Take screen screen sharing is easiest in two ways.
Sir, click on the blue button, the share
Okay.
screen,
Yeah, I've clicked on the blue button. Yeah.
and you can, it will be
I've
highlighted as
selected.
entire
I've
screen.
selected. Yeah. Now I've.
Yes, you will have to click in the inside a box, so it will again get highlighted.
Okay, it is there, highlighted. Is it now
Yes,
visible?
and
My
now,
screen is
sir, and you'll have to click on share button now. Again,
share
there will
button
be a share option, blue color button.
at the bottom. Is it? Okay. Okay.
Yes, sir. Yes, sir.
One second. That is hidden because of my taskbar. One second. How to share button? Yeah. Is it visible now? No. No.
No, sir, it is not
One
yet
second,
visible.
one second. There is I that is hidden. That is the reason. Okay, window. This. Yeah, I think now it is visible. No.
Yes, sir, it is now visible.
Okay. All right. Yeah. Yeah. Okay. Yeah. You can take this and this. Good morning to everybody who is participating in the national level Rusa sponsored webinar called Biofacet, organized by
So,
Patkar-Varde College,
sir,
University of
sorry to
Mumbai.
disturb you. Sir,
Yeah.
yes, your presentation is not visible in the presentation mode.
What to do? What to do?
Yes, sir.
What is
So on
the
the button which is just near to the negative sign, uh,
what should I do now?
sir, can you click on escape.
Escape. Okay.
Yes, sir.
Hmm. All right. Share. Shall I again start sharing once again?
Yes, sir.
Okay. I'll. Yeah. I'll start sharing once again. Share. Share screen. Window.
Sir, is it possible to present from my side?
Yeah, you can do that. That's the. I have already sent it to you.
Okay, sir.
Hmm. Okay. Am I visible? Your my I'm sharing my screen. No.
Sir, I'll share the screen from my side. It's
Okay.
fine.
Fine now. Shall I start now?
Sir, just give me 1 second.
Okay. Okay. Fine.
Sir, is it visible now?
Okay. What should I do?
No, sir. You will have to. You can just speak. You can explain. I'll change the slide from here.
All right. All right. Okay. No, this is. I'm not able to see the screen. This is visible now. Something choose to. Okay. Yeah. Yes. Yes. Okay. Thank you. Shall I start now?
Yes, sir. You may.
Okay. Sorry for this technical interruption. Welcome to this Rusa sponsored webinar called Biofacet, organized by Patkar-Varde College of University of Mumbai. First of all, I must thank all the organizers from Patkar-Varde College for inviting. Also, I would like to thank all the organizers who have invited me to be the resource person in this RUSA sponsored webinar. Especially, I must thank Dr. Ruchita Delvi, who has been in touch with me for a long period of time, and she has done a lot of background work in organizing this conference and this webinar. Thank you very much for each and everyone. And let us start with this topic called plant DNA barcoding. I request you to please change the slide. Next slide, please. Next slide, please. Okay. Let us first look into what is DNA barcoding. DNA barcoding is a method of species of different organisms. It may be plant species or animal species. The identification of the plant species or animal species or any organism is done by using a short section of DNA from a specific gene or genes. The premise of DNA barcoding is that by comparison with the reference library of DNA sections, an individual sequence can be used to uniquely identify an organism to the species level. I will tell you what is the reason for developing it and what are the advantages of this or what is the significance of DNA barcoding. Let us take a plant, a wild plant that you have discovered in the wild, that you have discovered in the forest. It may be a plant, it may be any other organism, it may be an animal, whatever it is. You want to identify it. Let us take the example of a plant because I am a botanist, I teach botany. Let us take a plant. All right. Recently, it happened to me. We found one plant in the Western Ghats, and the plant has been used for a long period of time as an anti-venomous plant. The fruits and the seeds of this plant are used by the tribals and the locals to treat the snake bites. It is known to be a very effective plant to treat the snake bites, but we didn't have any information about what is the name of this plant. It is called by so many common names, and I actually had no flowers at that time, only had seeds, only had fruits. I took these seeds and fruits to different people to identify this plant. There was there was no one who could identify this plant properly, because they asked me for the flowers, they asked me for the fresh flowers, they asked for the young flowers. Then only they said they can identify. That is one problem, and this problem can be solved later. I will tell you, by the help of DNA barcoding, it can be partly solved. The other problem that we may come across is, we'll buy so many herbal products. And you know very well these days, because of this Patanjali and Baba Ramdev, the herbal products have become very, very popular. So we buy the powder, seeds, plant materials for treating so many ailments. But when we buy these herbal products, whether it is a plant product or an animal product, whether it is a powder, whether it is a leaf, whether it is some other preparation, and what is the authenticity? How do you identify that a particular herbal product is genuinely collected from that plant? We have so many herbal products and plants, medicinal plants, which are very, very expensive. I'll just take the example of two such important plants which are very expensive and which are very popularly used. One is ginseng. Ginseng, the cost of ginseng root, the original wild ginseng, is about 1,000 US dollars in the market. If you are having wild ginseng, roots of ginseng, the cost is 1,000 US dollars. You want to buy and you want to use that ginseng roots, you buy it from the market. Okay, and how do you identify that this ginseng that you've got in bulk is original, it is not fake? How do you differentiate? And another example, I was just now talking to Dr. Giridhar about the saffron. The cost of saffron, original saffron, is also 1,000 to 2,000 dollars per pound. But how do you identify? Because the fake ones are there. I will show you the pictures later. The fake ones are there. And how do you exactly, precisely identify that this product is coming from the original plant? So there are so many problems, especially with regard to exact, correct identification of a plant species or an animal species. The idea is now that is available with us is plant DNA barcoding, or just called DNA barcoding. Now what we do here is, we create a DNA barcode for each and every plant, which is still being done. It is not done completely, it is partly done. The efforts are being taking place all over the world. So you can barcode a plant, in the sense the plant is converted into a simple barcode, and the barcode is available in a barcode library. There are so many libraries. One is bold, and you deposit the barcode that is in a barcode library. Next time when you want to identify the plant, what you do is isolate the DNA from the plant. It may not be from the plant. It may be from the environmental sample. It may be from the fruit. It may be from the leaf. It may be from any part of the plant. You isolate the DNA, all right, and then do barcoding and compare that with the reference one. All right, so you are not using the entire plant. You don't require a flower to identify the plant. You don't require fruits to identify the plant. We require only a small amount of material, plant material. Minimum requirement is about 2 grams of material is enough to identify a plant, and from that you isolate the DNA, and that DNA you convert into a barcode, and that barcode you compare with the reference barcode which is available in the database, and by the help of which you can identify the plant exactly without much difficulty, without the problems of understanding taxonomy at all. And that is what is the premise of DNA barcoding. I will tell you some of the important events and important steps, important procedures that are involved in the DNA barcoding in this presentation. We'll move on to the next slide. Can you change the slide, please? Next slide. Hello? Yeah. Now, this is the outline of DNA barcoding. So here we have plants or sampled. I'm concentrating on plant DNA barcoding. This can be applied to any other organism, but I'm concentrating only on plant DNA barcoding. So let us take the plant samples. You can take any plant sample. All right. I told you already you require only about 2 grams of the plant sample. From there, what you do is extract the DNA. How do you extract the DNA? There are so many methods by which you can extract DNA. And then you amplify the DNA and create a barcode. There are barcode creators, online barcode creators are available online. So you can convert the barcode, convert the DNA into a barcode after amplification by using PCR. And thus, what you're doing is the sequence of the plant that is collected from the plant is converted into a barcode. So this barcode is deposited in a barcode library, and that will become the reference. Next time, if anyone wants to identify the same plant, same thing they have to do. They have to isolate the DNA, they have to convert that into a barcode and compare that barcode with the barcode that is there in the reference library, so that the plant can be easily identified. That is the outline of DNA barcoding. We'll go to the next slide, please. All right. Little more elaboration is there here. We have the species. I told you it may be plant or animal. From there you isolate the DNA, and from that you can convert that into a barcode, and that barcode is deposited in the barcode libraries. There are so many DNA databases, barcode databases, where you are depositing. And I have given some examples of some barcodes which are already generated. So I have the example of Dendrobium barcode, which is there on the screen. Nvidia, you have Spyranthus, we have Telemitra, you have. These are the plant species of which you have created the barcode and which are available right now on the DNA barcode libraries. And now, this DNA barcoding is not a simple job, and it is not that anybody can do it. It is slightly tricky, and you require a lot of infrastructure for this. You require PCR, you require sequencers, and if you don't want to do all that, there are so many companies all over the world now, even in India, where the DNA barcode of a particular plant can be outsourced. You can give this job to some biotech company. They do it at the cost of 3,500 rupees per plant. The cost of DNA barcoding, if you have all the infrastructure. If you want to develop the infrastructure, it will be even higher. If you have the infrastructure, the cost of DNA barcoding will be about 3,500 rupees per plant. It can be even less than that. Okay, so that is what is the outline of plant DNA barcoding. We'll move to the next slide, please. Can we have the next slide? All right. Now, this is comparable to the supermarket scanners. I am telling you barcoding. So, if you go to a supermarket, all right, and each and every item that is kept in the supermarket has got a barcode. Earlier we had barcodes, now we have QR codes, and by using the barcodes, the customer can also identify the product. At the same time, even the stock manager or storekeeper also can identify the item. Similarly, plant DNA barcodes are also made in the same way for the known species which are kept in the library, and for the unknown species also we have to create that, you have to deposit in the library. All right, you can use parts of an organism, all right, to and make a barcode, or you can just do a simple catalog. Simply you can do catalog, and you can do it for as many plants as possible, and you can compare with the traditional taxonomy it's an effort to determine the species boundaries. So there are a lot of things that you can do. So now you can identify the plants, you can name the plant, all right, by not using your Linnaean taxonomy, not even going to the Kew Botanical Garden to compare your plant with the herbarium that is kept there in the Kew, and you can identify only by just scanning the barcode of the plant. This involves a lot of technology. That is what is the topic of this, that is what is the content of this topic, which I will explain later. But I'm just trying to give you some background about the plant DNA barcoding or just DNA barcoding. Please move the slide. All right, now I told you that you are doing DNA barcoding. We have different types of barcodes. See, the coding of a plant or an item need not be done only by using barcode. Barcode is one of the simple methods. There is QR code, there is Q code, there is short code. There are so many different methods. Right now, we are using only barcode. Maybe in the future, we can use different methods of coding. That is one thing. How do you do it? I have already told you. Isolate the DNA from the sample. So you require the plant sample or animal sample. All right. Amplify the target DNA. All right, and barcode it. And when you are amplifying, you have to use the PCR technology. You have to use polymerase chain reaction, by the help of which you amplify the DNA. All right, and then you again compare the resulting sequence against the reference that is there in the database, by the help of which you are matching the sequence. Now there is one step which is the link between barcode and the reference, that is converting your DNA sequence into a barcode. How do you convert your sequence into barcode? There are so many DNA barcode generators online. These are free tools that are available online, and one commonly used DNA barcode generator is Biorad. We have Biorad. What you do is you just go to Biorad, click Biorad on Google, you'll find Biorad, and you have a sequence. You just add the sequence, your barcode is created automatically. So you have to isolate the sequence, and then the sequence has to be barcoded. That is deposited into the reference library. And if you already have the reference sequence for that plant, you compare your barcode with that reference sequence, and thus you identify the plant species. Please change the slide. Yeah, this is what is done in case of a supermarket. You have a barcode. What is being scanned here is the QR code. So QR codes are scanned, which are available now on all the products that we buy in the market. And by scanning the QR code, you'll get the details about the product. All right, and similarly, barcodes are created for plants. You would just scan the barcodes and identify the plant species exactly to the species level. This is what is going to be the future. As of now, what we are doing is we are identifying the plants by taxonomic characters, which is a complex method of identifying the plants. But in future, if all the plants are barcoded and all the plants have got their reference barcodes in the library, then you don't have to worry about the identification at all. And at that time, at that point, taxonomy will become redundant. All right, you don't have to worry about taxonomy, need not worry about the characteristics of the plant. If you want to identify the plant, don't worry about complete history of and complete all paraphernalia of taxonomy. You just have to scan the barcode and identify the plant species. Now what we can do is we can do it only for the medicinally important, economically important plants. For that, we are creating barcodes. And for all these plants, you have already got the references in the databases. You can only do the barcoding and compare with the reference, and you will get the exact name of the plant species without any problem at all. But in taxonomy, you can always go wrong, and you can always identify the plant species wrongly. And if you want to identify the plant species, a lot of things are required, especially a flower is required, the plant, other details are also required. But for identifying a plant by using plant DNA barcoding method, all those things are not required. But you have to use the technology, and I'm going to tell you about the technology involved in plant DNA barcoding. Please change the slide. Okay, so this is what is happening here. And on the screen, you are seeing there are barcodes in a supermarket, and there is a scanner. The scanner has got a magnet. All right, the scanner reads the barcodes, and all the details that are hidden in the barcode are recorded in the scanner and displayed onto the monitor of your computer. And same thing is going to happen for each and every organism. All right, if it is barcoded. If it is barcoded, you can identify that with this. Maybe in the future, what I assume is all the herbal products, all right, let us say a herbal product has got 10 different plants. All right, and the manufacturer has used 10 different plants in a herbal product, and he is going to be compelled to provide the barcodes. And anyone who wants to see whether it is really that plant or not just have to scan that and identify the plant, or see that the plant that is used for the production of that herbal product is actually original. And if you have a doubt, if you get a doubt, you can also do that. Okay, because the technology is simple, and it is not a very complicated technology. It's a very simple technology. You can also do it. And if you can't do it, you can always all right, get it outsource it and get it done by the others, and you can definitely get to know about the exact name or exact identification of the plant that is used in the manufacturing of that herbal product. All right, please change the slide. Okay, now let us come to the technology. I've told you about what is barcoding. I think you've got some idea about what is plant DNA barcoding. Some of you may be knowing it. Now let us come to the technology. What is most important here is different gene regions are used to identify the different organismal groups for using barcodes. See, each and every plant or animal, all right, is unique because of its DNA. Besides the DNA that is there in the whole genome of the organism, there are certain regions which are very unique to that plant. All right, there may be many regions which are unique to us, but we are using only certain regions. For example, if you want to, all right, barcode a protist, a member of the kingdom Protista, lower organism, what generally we do is we use mitochondrial DNA. So in mitochondrial DNA, there are certain specific regions, and these regions are used as the identifying genes or regions of the plant. So the idea is very simple. Each and every plant or animal, all right, has got its DNA. The DNA of that organism is unique to it. All right, the entire DNA is unique, but at the same time, there are certain specific regions of the DNA which are very, very unique to it. And we are identifying the plant based on that unique region of the DNA that is there in that organism. It is something like DNA fingerprinting only. All right, but little elaboration and little different kind of technology we are using. So for the members of Protista, they are using mitochondrial DNA. Similarly, for you can also use rRNA, and for fungi they're using or you are using rRNA, and even for other organisms also you can use rRNA. But for plants you are using Rubisco genes. All right, these are unique genes which are available in some organisms, but there are certain other genes also are used. I will tell you about the other regions, but the summary is very simple. You are isolating only a unique regions of a particular gene, which is used as the identifying characteristic of that plant. That is the signature DNA section or region. That is what is used as the barcode. So you are not barcoding the entire genome. We are only barcoding a small section of DNA, which is about 400 to 800 bases, not more than that. So we are only barcoding that much of DNA, and that region of the DNA is very, very unique, and it doesn't show intraspecific variation. That means between the species of the same group, there is no variation in that region, but it shows significant variation in interspecific. Between two different species, it may show variations. So that is what is the important feature of DNA barcoding. So what you require is a section of DNA that is showing no variation or least amount of variation in the intraspecific, and shows variation in the interspecific regions. All right, please move the slide. All right. So these regions, as I told you already, having less intraspecific variation. Within the species, they should not show any variation, or they should show minimum variation. And they should show a lot of variation in the interspecific. Between two different species, there should be variation. All right. And this is what is known as barcoding gap. So the first term that I'm using next to barcoding is called barcoding gap. There are so many applications of DNA barcoding. I'll elaborate it later. But for the time being, all right, identification of plant leaves, even flowers, when flowers, fruits are not available. I had this difficulty, I just now told you. We could finally identify the plant species, the tree species, Barringtonia racemosa, on which we are working next. All right, so what is most important is this barcoding gap. That is, what is barcoding gap? Barcoding gap is there is least variation in the intraspecific and lot of variation in the interspecific. All right, and you can identify the plant without the flowers, without the fruits. What you require is only a small amount of the plant material. It may be powder, it may be fruit, or it may be anything. And you can even identify a plant by using its pollen grain. All right, and the pollen grain can be collected from the pollinating animals also. You have a bee which has got some pollen grains. All right, and you have got the pollen grains in the bee, and from that pollen grain also you can identify the plant, from which plant this bee has brought the honey. So this also can be identified. All right, that is called e-sample. I'll come to that later. All right, let's move the slide, please. Okay, now when when we are doing barcoding, there are two different ways, two different types of barcoding. One is the normal barcoding, the other one is called meta barcoding. All right, so when we are using barcoding for identifying an organism, sample DNA containing material can be collected from the organism. All right, the term meta barcoding is used when you're using, all right, large samples and a group of animals. Okay, and this is done especially All right, DNA metabarcoding is done especially in the diatom communities, in rivers and streams, in all right, which is used to assess the water quality. DNA barcoding techniques, all right, were initially developed by using 5' RNA. All right, I'll come to that later how it was developed. And we have two types. One is the regular barcoding, where the DNA sample is collected only from one organism or one particular organism. Second one is called DNA metabarcoding. What is DNA metabarcoding? Here we are using groups of organisms. All right, like for example, you collect the water sample, there are diatoms, all right, and you collect the diatoms. From there you isolate the DNA, and for that you do DNA barcoding, and this involves too many species of diatoms. But we want to know whether these diatoms are there or not, and if the diatoms are there, which group of diatoms are there, or if we have any other organisms which are contaminating the water. You know very well that there are a number of blue-green algae, all right, and these blue-green algae contaminate the water, and they cause a lot of problems. They produce some toxins, and if you want to identify the water quality, all right, you can do DNA metabarcoding, where groups of organisms are collected, and from there the DNA sample is collected. All right, yeah, move the slides please. All right. Now, I don't want to go into the detailed history of that, but I would like to mention the contribution of Paul Herbert. All right, the terminology, the modern DNA barcoding technology, and the standardization of the method, and identification of species, and potentially allocating unknown sequences to higher taxa. All the initial work, all right, all the primary work of DNA barcoding was done by Paul Herbert. We must thank him for developing this technology, because this is going a long way in the identification of plants on animals, and this is what is actually changing the status of very plant taxonomy or animal taxonomy. It will be completely different, and it will become much more simpler in the future. So it was all the methods and technologies which were initially developed by Paul Herbert. Please move the slide, please. Slide, please. Okay. Coming to the applications of DNA barcoding. So the people who can use this is taxonomists, especially this will become handy for the taxonomists. All right. And it will become easy for identification of the plants. Also, life scientists for identification of study of the organisms. Like you're working on a particular plant and you don't know the exact name of the plant. You want to know the exact name of the plant, and in that case, exact name can be identified and can be obtained by using DNA barcoding. And if you already know the name and you want to confirm whether it is the same plant or not, at that time also it will become useful. So taxonomists, life scientists use it, and conservationists. All right, there are so many plants which are now considered to be endangered, threatened, which are enlisted in the red data book, and these plants have to be conserved. And if you want to know whether the plant that you are considering is same or not, so they also can use DNA barcoding technique, and by the help of which they can identify the plant and they'll know the exact name of the plant. All right, and even environmental managers for the identification of harmful organisms or indicator species. There are so many plants which are actually causing problem as weeds, and they may be causing pathogenesis. They are causing so many diseases. So all these plants can be controlled, and before controlling, we have to identify the plant. And the identification of the plant can be done by using DNA barcoding. And there are so many indicator species. They may be pollution indicators, they may be environmental indicators, or they may be metal indicators. There are so many indicators of the plant, and these plant species also can be identified by using plant DNA barcoding, and especially legal. So when it comes to legal, especially in the forensics, if you want to identify the exact species name or exact plant, especially in forensics crime detection, then also you can use DNA barcoding, especially in case of wildlife crime, and some people are poaching the animals, the endangered animals, and they are carrying the parts of the animals. All right, and if you want to know exactly whether it is coming from which animal, and there also it can be used. And in legal trade of biodiversity, there are so many endangered plants and animals in the world which have to be conserved, and some of them are in very very small number, and they have to be conserved. And if you want to know the identification of that plant, then also it can be used. And in case of identification of species which are causing some diseases, there also you can use. Even in agriculture you can use in medicine, you can use in horticulture. So all these places you can use, all right, the DNA barcoding technology. Move the slide please. Slide. All right. Now coming to the exact premise, I was telling you that DNA barcoding is better as compared to our traditional plant taxonomy. You can see a plant that is there on the screen. The plant that you're seeing on the screen is Dioscorea alata. Like for example, if you discover this plant, you find this plant in the wild. The current method of identifying this plant is by our normal traditional taxonomy method, Linnaean taxonomy method, by binomial nomenclature. Raju. The current method by which you are identifying a plant, like for example, Dioscorea alata, what you're seeing on the screen, the current method of identification is based on a very complex and very, very complicated method. You can see it on the screen. You have to know the characteristics of leaves, roots, stem, flower, stigma, anther, corolla, calyx, floral diagram, floral formula. All, each and everything should be known. All right. Most importantly, the person who is trying to identify the plant should have this taxonomic knowledge, should know the meaning of these terms. All right, there are so many terms, technical terms, of which I sometimes I also get confused about the names. What is the meaning of those names? And when you want to identify, you have to enlist all these characters, which is a very complex process. Of course, it is a part of taxonomy, and that is what we are doing in taxonomy. So thus, the traditional Linnaean taxonomy is a very complex process, which is very subjective. And even with all this knowledge, with all the information that you have collected, still you may go wrong. You may identify the plant wrongly. All right. And let us come to plant DNA barcoding method. What we are doing is we are only collecting a small amount of DNA, small amount of gene, and we are barcoding it. And we are identifying the plant based on this DNA segment. All right, and that is what is the biggest advantage of plant DNA barcoding. So the traditional taxonomic method of identification of the plant is complex and it is subjective. You may go wrong at any point of time. At the same time, the plant DNA barcoding technology, where you can use this technology because it is simple, it is objective. Okay, so that is the comparison between our traditional method of identification of the plant and plant DNA barcoding method, which is the advanced technology, which is going to be the future of identification of the plant. Move the slide, please. All right. Now coming to the focus of this talk, and let us concentrate on one simple topic and one simple problem. I've just discovered one paper recently where a group of scientists published a paper in BMC Medicine. They worked on 44 herbal products from 12 different companies. One company that I can tell is Patanjali, we have Zandu, we have so many companies which are there in the world. Now they are producing herbal products. This is the fact. This is the survey, this is research done by a group of scientists. So they collected 44 samples produced by 12 different companies, and they found that, all right, out of 44, all right, 30, these are all from reputed companies, all right, were contaminated or substituted. So the consumers are put into risk. All right, the plants that are actually grown in the wild are collected from where the herbal products are produced. But these herbal companies, what they are doing is they are not actually bringing the original plant. I also conducted a survey in my lab. I brought the spirulina tablets from the market from different sources, and I studied what is there in the spirulina. I found that, all right, out of 12, all right, three groups, three spirulina tablets had acylatoria. Acylatoria is toxic. So, spirulina you are buying from the market thinking that there is spirulina inside. But if you see, if you take out and see, study it, and you don't have only spirulina, we have a mixture of different algae. And one dangerous algae that always is there as a contaminant in spirulina tablet is acylatoria. So, similar study was done by a group of scientists here. What they did is they collected 44 herbal products and they studied them, and these are produced by 12 very well known companies, and they found that out of 44, 30 products are having contaminations, substitutions. I will show you what are the contamination substitutions in the next slide. So we have alternate plant species are used as fillers, all right, and the saffron that you buy from market, all right, don't think that it is original sample. Always it can be fake. I will show you how the fake sample looks. And similarly, there are so many medicinal plants that are being used for the production of herbal products, and don't just go by the name of the company. They can be fakes, they can be substitutions, and they can be alternatives. So this is the purpose, this is the main focus, this is the main need. Because of this, all right, talk a little bit. This is the main need, main reason why I have taken this topic, so that everyone should become aware of this. Okay, so herbal products are not always are safe and always are not free from contaminants or substitutions. All right, let's go to the next slide, please. Next slide, please. Okay. Now, in the same study, what they did is I told you there are 44 plants, herbal product they studied. Okay. You can see that only some of them were authentic products. All right. And nearly 60% or 60 of them are incorrect product labels. That means, as I mentioned just now, spirulina should have been there, but we have all that acylatoria, incorrect product label. You are using some plant and some other name you are using. And there are so many places where you can see the substitution. And so many places you have contaminants. So many places we have fillers, and there are so many places there are no sequence at all. This is what is the need for DNA barcoding. All right, for fillers, for alternatives, for substitution, you can see the example of leafy vegetables that you buy from the market. Sometimes you buy methi or palak, all right, and in this methi and palak bundle there may be at least 2 or 3 parthenium plants, and if you don't know the difference between palak and methi and parthenium, you can use both, and parthenium is toxic. All right, so that is why barcoding, plant DNA barcoding is very, very important nowadays, especially for authentication of the medicinally important plants that are being used in the herbal products worldwide. Go to the next slide, please. Next slide, please. Okay, now I'm showing you on the screen the powders and also medicinal plants. So you can maybe somehow identify the plant. Like if I say tulsi, you may identify. Aloe vera, you may identify. But if aloe vera or tulsi or Lavulfia serpentina or Catharanthus roseus, if it is coming in the form of a powder, Can you identify? Even even herbal experts also may not be able to identify. But how can you identify it? Only based on the powder. All right, but the answer is you can do it by using plant DNA barcoding, and you don't require flowers, you don't require plant, you don't require anything. What you require is even a small amount of powder is enough. About 2 grams of powder is enough. From that you can isolate the DNA, and for that you can identify whether the plant is same or not. Don't worry, already many plants have been already barcoded and they are there in the databases. I will tell you, I will take you to the databases. Many plants have been already done. All right, nearly all right, about 5 lakh records are already available, and there are many more are coming in. And a lot of work is happening all over the world. A lot of data is getting poured in. So you can't identify by looking at the powder, but with the help of DNA barcoding, you can identify. Go to the next slide, please. All right. Now you can identify. Go to the next slide, please. Okay. I was telling you about the ginseng some time back. So ginseng is a plant which is available only in some parts of China naturally, all right, especially in North China. And it is highly medicinal because it is having anti-aging properties. All right, we know Japanese and Chinese are living longer, they look younger, all right, even at the olden ages, maybe because they're using ginseng. But the cost of original ginseng is 1,000 US dollars per pound. But you may get this root of ginseng. How do you know that it's original ginseng or not? If you're buying, let's say 10 grams, 5 grams, it's okay. But if you are a company, if you are a herbal product company, you are, let us say, buying 1 ton of ginseng from market, okay, you want to know whether the ginseng is original or not. How do you identify? If you go to a taxonomist, he can't identify by using roots. He wants flowers. Get the flowers, he will say. So identification is difficult. So what we can do is, by use of plant DNA barcoding technology, take only 2 grams of the material and go for barcoding method, use barcoding methods, and you can identify the plant. Next example. Go to the next slide, please. Next slide, please. Okay, now very commonly, all right, ginseng is contaminated. Plant products using herbal product using ginseng is contaminated with this fake plant. We have 2 plants on the screen. On the right hand side we have Panax ginseng, the original ginseng, and the left hand side we have, all right, Panax quinquefolia, which is a fake. And even for taxonomists, it becomes difficult for identification, differentiating, because these 2 plants look similar. Okay, and you want ginseng, all right, and this Panax quinquefolia is easily available, and they may give you. So how do you identify? Very difficult. So in this case, it's actually a crime that is happening all over the world. All right, and this is what is happening all over the world, where people are using fake plants and fake drugs, all right, and alternatives for highly valuable medicinal plants. Go to the next slide, please. I'll give you one more example. Saffron, all right, which is the original saffron, costs in the US market $5,000 per pound. It may be less in India because we grow in India, but in the world market, the original saffron is very expensive. Go to the next slide, please. Next slide, please. Okay, now I'm showing you the original saffron and also fake saffron. Okay, original saffron is from Crocus sativus, which grows in Kashmir, and the fake saffron is actually collected or is manufactured by using Cathamus tinctorius saffron, which is very cheap. Sometimes even silk threads are being used to fake the original saffron. Okay, they put color. This also looks originally same color. Only those who know the real nature of the original saffron can identify the difference. Otherwise, you can't. Okay, go to the next slide, please. Next slide, please. Okay, there is the difference between the original Rauwolfia serpentina, Sarpagandha, highly medicinal plant, and it is actually substituted by, all right, we have Ophiorhiza mungus, which is a fake one, which looks similar. If you don't know, they can fake it and they can give you this. Go to the next slide. Next slide, please. Okay, Saraka Ashoka, we have original Ashoka, which is Saraka Ashoka. All right, we have the fake Ashoka, and in many cases they replace original Saraka with the fake Saraka. All right, go to the next slide, please. Okay, now there's a big list of plants. All right, big list of medicinal plants. You are seeing this on the screen, and their medicinal value and their identification result. Okay, what is the original name of the plant? All right, and what is the substitute name result of the plant when they actually studied. So similar work is done by some other scientists. And the herbal products, okay, where they are supposed to use a particular plant, for example original ginseng, and they're using the alternate plant, and such works are done by a group of scientists. They've identified a number of plants where number of herbal products, innumerable number of products, herbal products, where, all right, the companies are using fake plants. And this is what is the fake plant. This is a big list. This is a big list. Please move the slide. Go to the next slide, please. Next slide, please. Next slide, please. I don't want to waste time on this. Next slide, please. These are all the plants already. DNA barcoding is done, and they are identified. This is a group of scientists who have reported this paper in the Nature journal. Okay, it is Nature Scientific Reports. Huge number of plants are being replaced by fake plants, and these fake plants are used in herbal products by the reputed companies. They are not used by the ordinary companies, they are used by the reputed companies. And that is the major problem with the exact identification of the plant, which is a must, and without which you can always go wrong. We can always, all right, end up in problems. Like I mentioned, we are using methi, and methi is contaminated by parthenium, and don't know the difference between methi and parthenium. Use parthenium, it can be toxic and it can be causing problems. So to avoid this, we can go for plant DNA barcoding. Move the slide, please. Okay. Now, coming to how do you do it, let us look into some of the methods used in DNA barcoding. All right. Now, we have three types of samples that are used. Three types of samples are used. One is the tissue sample. Okay. Go to the next slide. Next slide, please. Okay. There are three types of samples that are used in DNA barcoding. So the DNA barcoding, the first step is sampling, identification or collection of the sample. The three samples, one is tissue sample from the organism. You have a plant. From the plant, you require the tissue. You can take it from any part. Leaf is ideal. 2 grams of leaf is enough. That is one sample method. That is what is normally done. So normally what we do is tissue sample from any part of the plant or animal. Next is bulk sample. Okay, meta barcoding it's called, where you have a group of organisms, similar organism living in one place. Like I told you, in a water body there are diatoms living. You can take the bulk sample. Number 2. The third one is called environmental sample. What is environmental sample? Is also called e-sample. So here you are not having any plant at all. See, plants will leave some traces, or animals also will leave some traces in the soil. You are collecting the DNA from the soil or the environment. So it is a non-invasive method. It's also used in forensic technology. You have some samples that are left in some point of crime, and from there you are collecting the samples, where you are not collecting from the plant or animal directly. You are collecting it from the environment, okay, where the traces of the plant or animals are left. So that is called environmental sample. So three types of sources that can be used as samples for plant DNA barcoding or animal DNA barcoding or DNA barcoding as such. Go to next slide, please. So first step is sampling. And after sampling, the next step is, step number 2 is extraction of DNA. DNA extraction. I'll not go into these details of DNA extraction. That is not my focus. That is not the focus of this talk at all. So there are DNA extraction method. There are so many methods like phenylchloroform, isoamyl chloroform, protease K, CTAB method, spin column method, all right, or magnetic bead based method. So you can use any method of DNA extraction. Only thing is you need the DNA sample. You can see it on the screen. All right, spool of DNA fibrils are available there. So you get the DNA. So what you require is DNA. You extract the DNA from the sample. That is the second step. First, to identify the sample, collect the sample. It can collect it from 3 different sources. Next, you can use any DNA extraction method, extract DNA. Go to the next slide, please. Next slide, please. Okay, so the third step of the DNA barcoding is PCR. All right, so DNA barcoding requires the DNA that is in the sample that is extracted. There are several DNA extraction methods can be used. All right, factors like cost, time, sample type, yield effect, selection. So you can consider all these things and then use the method of DNA extraction that you want. So extraction is done. Then when DNA from the organismal or eDNA samples is amplified, you have to amplify the DNA by using PCR. And when you are doing PCR, there is one problem you can come across, or you can encounter some inhibitors, all right, and which can spoil your work. So you have to be careful about the inhibitors. And by avoiding these inhibitors, what you can do is you can use PCR technology, amplify the DNA, because you are collecting only a small amount of DNA and you have to amplify. That is the third step. First step is sample, second step is DNA extraction, the third step is PCR, amplify the DNA. Go to next slide, please. All right, now there are several criteria that are involved in the selection of the DNA region that can use as a barcode. I already mentioned in the beginning, you are doing DNA barcoding, all right, and you don't have to use the entire genome. You have to use only some amount of DNA and some region, specific region. Now, what are the criteria that you are using to identify the region or selecting the region? 3 criteria. One is discriminatory. All right, what is this? I already told you, unique for a species, identical for species members. Like for example, you have Barringtonia racemosa. All right, all the members of Barringtonia racemosa should have the same sequence, and there should not be any variation among the species. That is what is very important. So you should show minimum variation in the intraspecific. Okay, and next, universal. It should occur in all the species, all the members of that particular species, wherever that species is available. You collect a sample from India, all right, or Africa, wherever you collect it from, in all the plants, it should be there. It will be there, but you should see that it is there. And then robust. Okay, it can be amplified by using PCR. Using a small number of primers, you can amplify. You require primers for amplifying it. You have to develop primer for that particular sequence. Already done. All right, you have the information available in so many papers. You don't worry about that. So this is what is important in selecting the specific region. Go to next slide, please. Next slide. Okay, now I have three examples here of the sequences that you're selecting, which are good for you or which are bad for you. The first one is a sequence which is completely conserved. Okay, second one is no conservation, third one is partly conserved. Okay, now you have a sequence which is completely conserved, like for example, there are so many genes in plants and animals which are highly conserved. The H1 region, all right, of the H1 protein in DNA is highly conserved. Okay, it doesn't show variations, which cannot be used, such regions cannot be used. And if there is no conservation at all, which is showing variation even within the species, that also is not good at all. And what is ideal is a sequence which shows some conservation, which is good for PCR, good for barcoding. So sequences, when you're selecting, all right, for this study, you have to consider this. Go to the next slide, please. Okay, now you can see the work of CBAL. All right, CBAL is a community where they're working on DNA barcoding, and they have developed the database also. And they have come across and they have done a lot of studies. CBAL is Consortium for the Barcode of Life. They have recommended, all right, this is the final recommendation as of now. They have recommended, after considering all these things which I just now mentioned, okay, three important criteria, they've concluded that the chloroplast genes of RBCL and MATK, please remember, these genes are very specific to plants, and for plant DNA barcoding, two genes are used: RBCL gene, MATK genes. I'll tell you about these genes later. For animals, COI is used, but my focus is plants. So what we are doing is we are using the chloroplast genes. The genes that we are using is RBCL and MATK genes. You have to isolate these. These are, all right, suiting all the criteria that I mentioned some time back. Okay, go to next slide, please. Okay, now these are the genes that I mentioned. Okay, one is MATK. MATK is maturase K, which is about 1500 base pair, which are there in the plant chloroplast gene, and this encodes for organelle intron mutase, a protein that splices group 2 introns. That is the job of this gene, and this gene is unique to a particular species. This is not all of a sudden we have to come to this conclusion. Over a lot of time, people have worked and they have come to this conclusion. As of now, this is one of the best genes. The other gene is RBCL gene, which is about 1400 base pairs located on the large on the chloroplast genome, and this also has got a very good. It actually is very good for DNA barcoding, plant DNA barcoding, because it actually, all right, compiles to, or it also answers all the problems of your selection of the region, which I mentioned some time back. All right, and this is called barcode gap, and the barcode gap is very important, and these two genes are used, and I've given you the structures of these, the location of these genes here on the screen. Go to the next slide, please. Okay, and after selecting the genes, after selecting the specific region, after isolating the gene, the next step is PCR, polymerase chain reaction, where you are amplifying the DNA. I'm not going to the details of that. Go to the next slide, please. Okay, so this is what is a this is what is a PCR technology, polymerase chain reaction. I'll not go into these details, so you can amplify the DNA by using PCR technology. All right, go to the next slide, please. Okay, so amplification of DNA, extracted DNA, is required as a step in DNA barcoding. Typically, a small fragment of DNA material is used, and the sequence length is also about 400 to 800 base pairs. All right, amplification of eDNA, where the amount of DNA is very less, is even more important, where the size of the DNA sample is even lesser. It is less than 200 base space. So whenever you are using eDNA, the sequence length is lesser than the normal. So the sequence that you're using is very, very small, and the amount of DNA that you have got also is very small. That is why the DNA has to be amplified. Okay, next slide, please. Okay, and the next step after PCR is sequencing. So you've collected the sample, you've extracted the DNA, you amplified the DNA. The next step is sequencing. You want to know the sequences. Now, earlier the sequencing was very, very difficult and very, very cumbersome process. The first sequence that was developed was kept in a place where it was about 1,200 square foot area. But now the sequencers have become so handy. That means that they are so small. You can see on the right hand side, a man is holding a sequencer in his hand. You can keep it in your pocket. A DNA sequencer is as small as your mobile phone. You can keep it in your pocket. Very simple it is. So you can do sequencing. There are so many methods of DNA sequencing. All right, I will not go into the details of that. All right, I will just show you how sequencing is done. So you do the sequencing of the DNA sample that is extracted. Go to next slide, please. Okay, so this is what is Oxford millipore DNA sequencer. All right, go to next slide, please. Next slide, please. Okay, so this is how sequencing is done now. Earlier sequencing, all right, you can read the history of sequencing. Okay, there are so many methods about you have, starting from Sanger's method. There are so many methods of DNA sequencing, but as of now, what we are using this millipore technology, all right, where the DNA fibril will enter through a pore, and as the DNA is entering, passing, there is a scanner. The scanner will read each and every base, and based on the response that is coming out from each sequence, each base, whether it is adenine, thymine, cytosine, guanine, it is reflected on the monitor. It is a very, very fast process. You can do sequencing it very, very fast. So it is very easy, very simple, and very very cost effective also. The DNA sequencing has gone into a revolution. When DNA Human Genome Project was completed, the human genome project cost about 3 billion US dollars. All right, so that means cost of genome sequencing was $3 billion. Now, at present, the cost of sequencing of one human genome is, or one genome, complete genome, is about $1,000. $3 billion, it has come to $1,000. So, sequencing has gone through a revolution where it has become very, very cheap now. What you're seeing is millipore sequencer, where you can sequence the DNA that you have selected. The sequence goes into the pore, and as it is going into the pore, what you can see, the red color structure. The red color structure is the scanner. It scans each and every base. Adenine, it reflects on the screen. Thymine, it reflects on the screen. Guanine, it reflects on the screen. So as the DNA is passing, you can see the sequences appearing on your TV monitor. So you can do it live. You can sequence it live. You can see the sequence happening in the millipore sequencer live on the screen. So sequencing can be done in a very, very simple way now. If you don't have a sequencer in your lab, you can always outsource it, get it done from the companies. People are there who can do your DNA sequencing and give you the results. Go to the next slide, please. So that is the penultimate step in plant DNA barcoding. All right. Now we are using markers. All right. And the markers, I've already told you about the markers. All right. And what is most important in marker is the barcode gap. Go to next slide, please. This I've already told you. Next slide, please. All right. Go to next slide, please. Same. I don't want to repeat this again. All right. This is the markers. Okay. Go to next slide, please. All right. Now, I've already told you about this. The genes that we're using is MATK is one. All right. And in addition to MATK, and RBCL. TRNH also is being used nowadays. So the three important regions that are very unique, and they actually will suit for plant DNA barcoding. They are being used, and they're all coming from the chloroplasts of the plants. This is good for plant DNA barcoding. Next slide. Okay. Now the last step is called reference library. Okay, now you have done the DNA barcoding. All right. And how do you identify by using DNA barcoding? You have what is known as reference libraries, which are used for taxonomic identification. All right. And these databases contain DNA barcodes assigned to specifically previously identified taxa. Already nearly 5 lakh sequences or plant species have been identified. Sequences are available in the databases. Okay, and that is happening. So the DNA barcoding, final step of DNA barcoding is done in a reference library. What is a reference library? Reference library is nothing but DNA barcoding database. All right, there are databases which are actually constructed by the deposition of DNA barcoding done by different people, where DNA barcoding is done and it is deposited in the barcoding libraries. All right, and this is having collaboration with the other institutes like taxonomical institutions for identification, for accurate identification. Okay, so the last step is you have to build the reference library, which is being built. There are so many libraries. If you are doing it for the first time, deposit it in the DNA reference library, or if you want to check whether your plant is already done and you want to compare, you can always do that. You can do two things in the reference libraries. One, you can build the library. You can do a barcoding of a plant, deposit in the library. We are working on one plant, Bantonia racemosa. All right. Or if you want to identify plant which is already done by somebody, you can always do it. You can always compare that the existing. And this process is online. Okay. These databases are online. You can do it. You can get your result within seconds. Go to the next slide, please. Next slide, please. Now, there are so many libraries. The largest DNA barcoding library, International Barcode of Life Project, IBOL. All right. The second largest is called BOLD. Okay. Second largest, which has got 5 lakh bins. These are called bins. Bins are nothing but barcode index numbers. The third biggest one is Unite. There is a database called Unite. The fourth one is Diet Barcode Database. There are so many plant DNA barcoding databases. I have only mentioned 4 important ones. The biggest one is IBOL. Second one is Bold. Third one is Unite. So these are there available online. You can always Google and find out. Go to these and see what data is available. If you want, you can deposit the data of DNA barcoding already done by you, or you can compare the one which is already done there, and when you want to identify a particular plant species to its exact species location. Go to the next slide, please. Next slide, please. Okay, this is the Bold Barcode of Life database, IBOL, Bold systems, the home page of that. Next slide, please. Okay, this is Barcode of Life database, that is Bold. You can explore the data. This is the home page of that, which I've taken recently. All right, this is the recent version V4. Go to the next slide, please. Okay, now I have tried to identify the sequence of Oryza sativa, which I did it recently. So Oryza sativa is one plant where a lot of work has been done. You can see here there are about 210 records of one species, because they say there are about 5,000 varieties of rice in the world. So 210 records are there for Oryza sativa. But all of them are having the same sequence with reference to barcoding sequence. Okay, I have worked on that. You can see here. The next slide, please. Next slide, please. This is the FASTA sequence of the Oryza sativa RBCL sequence. This is the sequence, original sequence, which I've collected from board only. This is how it looks in the FASTA format. Okay. Next slide, please. And this is another database called Unite. All right. You can go to Google and search for these databases. They are freely available. Next slide, please. Next slide, please. This is one more diet barcode. All right. This is also open access, freely available. Okay. Where you can find the database, the barcode, the diatoms, and all the other organisms. Next slide, please. Okay. Now, the last step, last part of it is what is bioinformatics analysis. So how does this identification is done by the interface between you and the database? All right. Now, how is it done? In order to obtain well-structured, clean, and interpretable data, sequencing must be processed by using bioinformatics analysis. All right. What you require is the sample that is in the form of a FASTA format, and this sequence, all right, is what is loaded as a source material for obtaining the DNA barcode, and for doing this interface between you as a plant taxonomist and the bioinformatics, where you have a database. What is most important is the bioinformatics analysis. Now we have a sequence. All right. And how is this sequence compared with the existing sequence, and see whether the sequence is related or not? You have a sequence, and the sequence is compared with the reference sequence, and you want to identify these sequences are same or not. How to do it? And this is done by bioinformatics analysis. It's an elaborate step. It's another subject altogether. It's bioinformatics. There are a lot of things that are involved in this. I'll not go into the details of that. I've made several videos on bioinformatics. You can go to my YouTube channel and read. You can see those things, and if you want, you can learn. So it is another realm that is involved in plant DNA barcoding, but you don't have to do that. You don't have to do that. What you have to do is just collect the sequence. That is enough, and convert that into barcode. Compare that with the reference sequence. If you want to do that, you can do that also. Go to the next slide, please. Okay, now the identification is done by what is known as sequence similarity search. You have a sequence, the sequence has to be compared with the reference sequence, and this comparison is done by what is known as sequence similarity search. And this is done by a tool called BLAST, Basic Local Alignment Search Tool, which is also freely available online. If you have a sequence, you can always compare your sequence with the reference sequence by using this tool called BLAST. This I told you already, it is a part of bioinformatics. I'll not go into the details of this. If you want, you can learn more about it. You can watch my videos, and there are so many other sources also, but from the help by using those sources, you can learn them. So what is actually done is, for comparison, BLAST is used. Okay, go to next slide, please. Okay, this BLAST, this is the home page of BLAST. Okay, I've made several videos on bioinformatics. You can go to my YouTube channel. It is in my name only. You can just type in Google Dr. Jairam Reddy YouTube channel. You can go to my channel. I have more than 210 videos already there. Okay, and I have a video also on BLAST practical and other bioinformatics practical. All the different practicals of bioinformatics I've already made videos. You can actually, I teach bioinformatics in my college, so you can go to the YouTube and you can watch these videos if you want more information about how BLAST is done and what is the technology involved in this and how comparison is done. Okay, go to the next slide, please. Okay, finally, the applications of plant DNA barcoding. One is identification of species. Next is detection of invasive species. All right, delimiting the cryptic species. There are so many species which are identified as one species, and actually their name is different. Okay, there is a lot of ambiguity, confusion. You can delimit, you can rename the plant. So by using DNA barcoding, you have an opportunity to rename a plant species, definitely. All right, which are there in the fringe, which are there in the hedge, which are there in the margin. All right, if they are not sure about whether they are this species or that species, you can always use DNA barcoding and specifically identify. So delimiting the cryptic species is one thing which you can do. All right, and with by doing this you can publish a very good original paper. You can do diet analysis, food web application, especially barcoding can be done for food safety, bio and monitoring, ecological assessment. All right, I also told you what you can do is you can do it in case of medicinal plants, herbal plants, which are widely being used these days. So for exact and precise identification of that, you can use plant DNA barcoding. Next slide, please. Okay, now this is the overall summary. I've just summarized it here. All right, I need not go through it once again. Next slide, please. Okay, this is the examples of some of the DNA barcoding, and they can be converted into QR code also. Next slide, please. Summary again. Okay, summary again. Take the sample, extract DNA. All right, so summary again. Coming to the limitations, last part of it, it is always not true that the intraspecific variability is negligible. All right, and the least lower than the interspecific. There is no universal DNA barcode gene. So you cannot have an universal DNA barcoding gene. Barcode sequences should be generated from type specimens, thus rely on classical taxonomy. They also rely on taxonomy. So you need to have taxonomy. You should have the taxonomy first. And this can be done only for the plants that are already taxonomically identified, taxonomically identified. For that only you can do it. You can't replace taxonomy. Taxonomy should be there, but already identified plant, you need not again identify by that method. You can use some other method of identification. So these are the limitations. Next slide, please. All right, so that's it from my side. Thank you very much for listening to me patiently. And I must thank each and every one of you. I must especially thank the delegates for bearing with me, especially initially when there was some technical problems. I must thank all the organizers of this Rusa webinar conducted by Patkar-Varde College of Mumbai University. Thank you for giving me this opportunity for uh, being the resource person of this Rusa webinar. Okay, so with this I conclude. Over to Tanvi or Priyanka.
Okay, thank you, sir. We'll move on to the question answer session. We have a question from Devanshi. She wants you to explain more about metabarcoding and analysis of water.
Okay. See, barcoding is one method where you are using one particular organism. For example, you want to identify Rauwolfia serpentina or Panax ginseng. You identify the plant by taking some sample from it and do the entire process. I will not repeat that process. And then do the barcoding and identify whether the plant is either exactly Panax or something else. That is what has been done already by so many scientists. I've given a big list where it has been done. Coming to meta barcoding, so here it is done especially in the environmental studies. Okay, now what they want to do is they don't want to identify, their intention is not identifying one particular plant species. They want to know whether there are diatoms or not in a water body. For that, what they do is they collect the samples, they collect the entire DNA, and there is a barcode for diatoms. Not one particular species. Diatom is one group of plants. Okay, dinoflagellates, we call them as one kind of algae like dinoflagellates. We have diatoms is one kind of algae, where these diatoms have some unique identification as a group. As a group, they have a unique identification. All right. Now, by doing meta-DNA barcoding, you will know whether the items are not there, or not. If the items are there, it is okay. The water is healthy. But if you have, let's say, some blue-green algae, let's say Acylatoria or some other toxic algae, which is not good for a good water body, not good even for the fish. Okay, so you want to know whether diatoms are there or blue-green algae are there. So here it is group identification, not species identification. So plant DNA barcoding is species identification. You're identifying a particular species. Metabar DNA barcoding is identification of a group of species, especially in when we have a group which are having similar characteristics. I hope I've answered your question.
Okay, thank you, sir. Next question is from Giridhar, sir. Are there any FSSAI guidelines for herbal drugs for barcoding?
As of now, there are no guidelines as such, okay, because it is still in the preliminary stages. There are no guidelines as such now. That is what I am proposing. Now, in the future, if any herbal company, all right, produces a herbal product like maybe ginseng or maybe saffron, I am recommending, I am writing a letter, I am recommending that they must complete the DNA barcoding, and they must deposit the DNA barcoding in the library, and they must provide the DNA barcoding on the product. Okay, these guidelines are not formulated so far. They have to be formulated, because you are also having, you can also use your good offices, you can also promote this, you can also propagate this message where herbal products must carry DNA barcoding, which is not happening anywhere in the world now. It should happen. So you can also use this platform. Maybe you can use your good office. Maybe you can also write some
because
code, isn't it, sir? You're also working
because
on food. Yes, sir.
yeah, because our you know that CFTR is a signpost for spirulina research.
Yes,
So
sir.
yeah, I surprised to see to know that it is adulterated with oscillatoria,
Yes, sir. I
because
did it in
many
my lab.
firms. Yeah, is it? Have you analyzed all the commercial brands like Paris, Nova, Dabur, or like that,
No,
or
I
it is a
didn't do that.
local
It's
groups?
only a term paper. We
Oh,
are graduate student, do some small work. I didn't actually should have taken it further. I didn't do further work.
yes, sir. Yeah,
Which
it
is
is really, you must do that one. It will really
yes, sir.
helpful.
See, I brought spirulina from you, sir. Chandramohan brought spirulina from your lab, and we were culturing it here. So to comparison, sir. You remember Chandramohan,
Oh,
sir?
okay.
N.
Yes,
Chandramohan.
yes, yes. Yeah,
He
I
finished his
remember.
PhD recently. So he brought
Oh, okay.
spirulina from CFTRA only. We were culturing it in our lab for comparison. We were culturing it here.
Oh,
But this can be taken
okay.
up. See, we have
Yeah, yeah.
limitations because we don't have much infrastructure in colleges. One problem. The other problem is we don't have much time also, like you. You're all dedicated scientists. But we are not. We are amateur scientists. We don't have much time. We have to
Not like that.
we
Your
have to
talk,
do a lot of other works also. We have
no,
a
but
lot of
your talk
clerical
is
work.
excellent. Your talk
Thank
is
you
an
very much.
excellent, and it has been a very highly informative. Very highly informative.
Thank you, sir.
Okay, thank you.
Thank you, sir. Thank you, sir.
Thank you. Just a second. Sorry, we were having a technical problem. I think that is all the questions that we had. So, now to conclude the session, I request Mr. Pushkar Bhojane to extend a hearty vote of thanks.
Thank you, Tanvi ma'am. I, Mr. Pushkar Bhojne, on behalf of Patkar-Varde College and the entire faculty of Department of Microbiology and Biotechnology, deem it a great honor to propose a vote of thanks to all those who made this webinar a resounding one. I will extend a very hearty vote of thanks to both the speakers for taking out their precious time for enlightening participants and making this webinar a great success. I will take this opportunity to thank Dr. Giridhar Parvartan sir for his efforts towards advances in tissue culture technology for enhancement of food value. And will also like to acknowledge my gratitude to Jairam Redisa sir for his enormous efforts towards plant DNA barcodes. I would like to express a profound gratitude to our CEO, Dr. Mala Thakur, Principal Dr. Shrikant Sawant, Chief Coordinator Mrs. Suchita Talvi, and co-coordinators Mrs. Anupama Tomar and Mrs. Neha Kolasu for their moral support and guidance. We are also grateful to our sponsoring organization, Rusa. This event wouldn't have been successful without the constant support from our staff members and wouldn't have run smoothly without a technical team. Last but not the least, I also thank the wonderful participants who have joined with us from all over the country and for their active participation throughout the webinar. The link for the feedback form will be posted in the chat box. Once again, thanks to all for their cordial cooperation. Stay home, stay safe.
Thank you, Tanvi.
Yes, thank you, sir.
Okay, I'll log out now. Okay, thank you very much.
Yes, sir.
Okay, thank you.